Hello, good morning.
Good morning, sir.
Good morning.
How are you?
Please, we are good.
Where are the rest? I'm supposed to meet over 40 students. I have about 16 students here, so I think let me still give you about 3 minutes to contact the rest. So what we are going to do, I don't want to leave anyone behind. So you can contact your friends, share the link with them, and probably 5 minutes time we can start our class. All right, good morning once again. Let's see, probably if you can start, then your, your colleagues may join later. I hope you can hear me.
Please, yes.
All right. So basically, last week we started with this particular course, food biochemistry, where our focus last week was looking at what food biochemistry was and how they are important in our field as food and nutrition students, or people that we are going to find ourselves in the food and processing industries. Then basically, we started looking at some portion of genetics, whereby we spoke about, look at DNA as a main topic that we look at last week. In the specific one that the specific thing that we look at was DNA. We spoke about genome, and how individuals inherit this a specific trait from our parents, and how this trait is being inherited. So we even look at the structure of the DNA itself. I think I gave you an assignment, right? Did I give you an assignment?
Yes, sir. Yes.
And what was the question? Let me look at it before probably I proceed. What was the question?
So you asked us to look out for the function of the nucleotide.
Okay, the trinucleotide, right? Yes, I remember I told you to look for the function of the trinucleotide. And the nucleotide, the components of this nucleotide are the pentose sugar. If you remember, I told you people that under normal circumstances, all sugar should have 6 carbon. But when it comes to DNA and RNA, their sugar is 5. That is what we call the pentose sugar. Then we also have what we call the nitrogenous base, which is also a component of the nucleotide. We spoke about the phosphate group. So these are the 3 nucleotides that I made mention of. And I asked you people to find the functions. And I think through my presentation last week, I think some of the functions were mentioned. So those that you did the assignment that I gave you, I think I want to get my solution now. And I'm expecting everybody to do it. So I can just go through the chat box and just mention anybody's name at all. Then you tell me what you did. So those of you that you want to willingly share your solution with us, I think you are welcome. So at this point, I welcome anyone who want to share. Anyone who want to tell me the solution? Oh, you guys didn't do it. You know, by now someone has opened ChatGPT. Please, I'm waiting. Ask me if no one is talking. Your name is just on my screen. Oh. Ask me and Efia. There are 2 people that I have them on my screen before I move to other. Ask me, can you tell me something that you read on these 3 nucleotides and their function when it comes to DNA or RNA? Please, I hope you can hear me.
Sir, yes. Sir, can we say, I'm trying.
I want you to try it.
Sir, it helps in the structure formation of the pentose sugar.
What are you talking of? I think it should be
The
specific.
pentose sugar. Sir,
pentose
sugar. Yes.
Okay.
It helps in the structure formation of the pentose sugar.
let's say, so sugar, we know that sugar is a carbohydrate base. I'm using my mind to say sugar, the end product of carbohydrate is sugar, that is the glucose. So I will say that it helps to break down the carbohydrate, like the energy component in the system.
Yeah, I'm confused. Oh.
So I'm saying that. So this I'm using my mind.
And don't use your
Penta-
don't use your mind. We want to speak to facts. Because we are in science class, if we use our mind, we will turn our class to social sciences class. So in science, everything is supposed to be empirically backed. So you can use your mind.
yes,
When you
sir.
are doing your social studies and your family management, then use your mind to write up. But in science class, remember that.
Okay.
Can
So
someone
I'm
also
saying
try?
so I'm saying that because pentose sugar, eh, when we say pentose sugar, because we are talking about the sugars, it helps in energy metabolism.
I think you are moving far away from DNA and probably you are talking about something different. What you are talking is
No,
also
sir.
some
I'm talking
aspect
about
of
the pentose sugar, the components. You said is three.
just the other. You all of
Yes,
you are quiet and she is confusing us. You don't want to talk. Yes, continue confusing us. We are listening.
so I'm saying that when you take the pentose sugar, when you in the DNA and then the RNA, it helps to if I'm saying it helps to build the energy structure in the system.
Okay. Let me just listen to. I want to also listen to others and come back to you.
Yes, sir. Yes,
Uh,
sir.
please on your microphone and talk to me. I gave you an assignment. It's a week now, so I'm expecting you to stand by now. If I realize that when I give you an oral assignment, you don't do it, probably I will start giving you the one that you will submit it. I will give you my email, then you send the assignment to me so that I will have a look at it. It seems this one, most of you didn't do. And now that I realize you people didn't do, I won't talk about it. I will leave it like that. So maybe when you meet it in an exams, you find a way to tell us. Is anyone ready to talk? The function of the three nucleotide that we made mention of. Is anyone ready to talk? Today, since I'm teaching in a cemetery, let me see. Evelyn, is Evelyn here? Evelyn, I think I have only one Evelyn. So if I'm not
Yes,
able
sir.
to,
Yes, sir, please, I'm here.
Evelyn. Yes, last week I remember your voice. I think you were talking a lot. What did you read about it?
Sir, please, what I also read was about the phosphate group, the function of the phosphate group.
Hmm,
Um, and he said they store energy.
I'm listening. Continue. Is that the only
So
thing you read?
they help to store energy and also helping the transfer.
Transfer of
I didn't go detailed, sir, please.
info detail. Okay. Any other person? Efia. If I mention your name, you
So,
don't respond. It means if I mention your name, you don't respond. It means you have joined the line. You are still sleeping. Yes, Efia.
no.
What did you read about it?
So, me, what I did was I went through the structure of the nucleotide. I wasn't specific. So, what I wrote down was they are the building blocks of the DNA and RNA. And then they are the fundamental units that make up the DNA and the RNA. And they are the molecules that store and transmit genetic information. And they also help in growth and development. That they are involved in essential biological processes like growth and development. and cell division. And also, they store and transfer energy in the various cellular processes. That was what I wrote.
Okay, when I look at your responses, I think you did a general reading on the nucleotide as a whole.
Yes, yes.
I gave you the specific nucleotide. There are 3, and I was expecting you guys to tell me the specific function that each of the nucleotide perform. When we say pentose sugar, what does pentose sugar do when it comes to DNA? What does phosphate group does when it comes to DNA? And also, what does the nitrogenous base does when it comes to probably the DNA formation? All right. So, looking at most of your responses, let me just say this so that you can do your further reading and just come out with the phosphate group mostly involved in the structural formation. Okay, it's mostly involved in the structural formation. That is the key thing that I will talk about here when we talk of the phosphate group. And also the pentose sugar. I think most of you had the pentose sugar also help the phosphate group to attach itself to the nitrogenous base. That is one. It also gives direction. Remember last week I was talking about 5 to 3. That I said that they move in anti-parallel direction. So when it is anti-parallel, when here is 1 and the other end, let's say it contains about 5. Let's say one particular DNA contains about, let's say, 5 different proteins, 1 to 5. So when you have 2 strands of DNA, remember I made mention that DNA is made up of 2. They are double-stranded as compared to RNA, which is single-stranded. If you remember that particular point that I made the other time. So if we have a DNA that is made up of 5 amino acid units, let me put it that way, and it is double-stranded, it means that each of the strands is made up of 5. So what we are saying is the first strand will move from 1 to 5, but the second strand will move from 5 to 1. And that is why I made mention that they are anti-parallel. They wind to each other. But when it is parallel, it does not. When you say anything is parallel, 2 parallel lines, they move in a straight line without meeting. But this one is anti-parallel, so they meet at a point in time. And how they meet is, we call it a 5 to 3 dimensional. This point is not all that what is it important when it comes to your area. Just that I just want you to have a few understanding of that. Then we can also talk of the nitrogenous base, and I told you the nitrogenous base is the part that contains the specific genetic code. If you remember, the nitrogenous base, it is the part that contains the specific genetic code, that code for specific protein. So the nitrogenous base specifically, we can talk of the the thymine, the guanine, the cytosine, and the rest. These are the bases, and these bases code for specific protein. So when you talk of when you pick something like let me see if I have, when you pick something like let me say valine. Valine is an essential amino acid. It's a specific essential amino acid, which the code for it is cytosine, guanine, and adenine. So this particular code will be carried by the nitrogenous base, and when it codes for that, it will communicate to the ribosome that this is a specific protein that the body want to produce. So put it in mind that with the nitrogenous base, it involved with the genetic coding. It involved with the genetic coding or the base pairing, if you remember that. It also carries genetic information. So you can read more. Their functions are many. So you can read more on that and possibly you will get to understand more. But don't forget that particular thing, that those nucleotides, they are very, very, very important when it comes to DNA, issue of learning about DNA and RNA. Without a proper understanding of them, probably it will be very difficult for you to know what they are. So I don't know whether you have any questions from last week that you want me to address it before possibly I move on. Yes, me. I can see your hand up.
Sir, please, the pentose sugar. You said they help the phosphate group to do what?
The pentose sugar. I said that
Yeah.
it linked the phosphate group and that of the nitrogenous group to the carbon itself, the nitrogenous base. I hope you understand. It helped in the linkage. You know, I don't know whether you know, I gave you a particular structure.
Yes.
Let me see if I can find it here. I gave you a structure whereby the nitrogenous base, the pentose sugar, and that of a phosphate group are linked together. So the linkage is facilitated by the pentose sugar. That is what I mean.
Okay, sir. Thank you.
You're welcome.
Please, sir,
Yes.
you also made mention of it helped to give direction to what I didn't get that place right.
So I was saying that when it comes to DNA or RNA formation, there's something that we call replication and transcription. I think we will do that one today. Probably when we are done with that particular one, you get that particular understanding. The pentose sugar is also involved in replication and transcription of genes. So we will talk about it today. So probably you can note that question down. When we are done here, still not clear, then probably I can.
Thank you, sir.
Any other? The rest, are you okay with the first unit that we did last week? Okay. I think in our basic communication, you say that silence means consent, right? In our basic communication, you say silence means consent. So let me quickly move on to what we are going to do today. Today, our focus is going to be on DNA replication. DNA replication. Remember last week, I told you people that RNA is mostly synthesized from DNA. And anytime you want to synthesize a specific proteins, we need DNA must be converted into RNA or must be copied in the form of RNA. And I made that specific RNA is called the messenger RNA. And I told you DNA is mostly found in the nucleus. DNA cannot be activated outside the nucleus. So always DNA is found inside the nucleus of a cell. But protein synthesis does not occur in the nucleus of a cell. So protein synthesis occurs at which part of the cell? I made mention of it last week. Which part of the cell does protein synthesis occur? Anyone? Yes.
Ribotamine or something like that.
Ribotamine. I want someone to put it in a proper context. I think you have an idea. Yes, protein synthesis occurs in which part of the cell? It's Ula around Ula. It's Ula here. Yes, protein synthesis. Which part of the cell does protein synthesis occur? Hey, my people, it means you guys are not reading the slides I sent to you last week. But this is basic question that I'm expecting you to know. Okay. Remember I told you that with the DNA, it is mostly found inside our nucleus. But when it comes to issue of RNA, when it comes to issue of protein synthesis, RNA cannot, or protein synthesis cannot occur in the nucleus. It rather occurs in part of the cell that we call the ribosome. So there is a specific, specific RNA that is involved in the protein synthesis in the RNA. And I said that one is called the ribosomal RNA. So it is telling you that is the RNA that is found in the ribosome. That is why it got its name ribosomal RNA. Last week we looked at 3 types of RNA. We spoke about the messenger RNA that picked the coding from the nucleus and bring it to the cytoplasm. Then we have what we call the transfer RNA transfer the code or the messenger RNA, or which carry the code to that of the ribosome, and the ribosomal RNA will cause the synthesis of the protein to occur. So these are the basic things that I'm expecting you to understand them as at now. If not, as we are moving forward, it is going to get complicated, and I must be sincere. I know most of these things are not common or familiar with you. So if you leave the slides down, so you are going to write quizzes or exams before you write them, you read them, it will be very difficult for you to get the concept. Very, very difficult. I have to be very sincere. Biochemistry is not something that you can leave it and read it when you are going to write an exam, because you need to get the basis. And the questions are not going to be still the types of RNA that we have. No, it is going to be a form of application. So if you don't join lectures and you don't read your notes and ask questions, it will be very difficult when it comes to answering of such questions. So I'm begging you, I'm begging you today. If you don't listen, you will come and beg me tomorrow. All right. So
please, sir. We've not gotten the slide yet, too. That is making it difficult for us.
As at 1 week, you've not gotten the slides and no one has made an effort to contact me, then I think the fault is not coming from me. Is that not so? As a student, you should be running and finding ways to get your slides. I asked you guys to form a group, and per my knowledge, the group was formed, and I personally sent the slides inside that particular platform. So if you don't find yourself in that particular platform, yes,
so
the slides on the platform.
They are on the platform. So if that person who doesn't have slides,
so if you are a student and I ask you,
then some of us are not on the platform.
So,
please, we gave an avenue. We gave an avenue to contact you.
What I made mention the other time was create a platform, add me to the platform. Any information that I get, even aside the lecture slides, I will forward it to the platform so that you can use it as students. I was there, I saw a link. Me personally, when you add me to platform, it will not work unless you send me a link, then I will join the platform myself. I joined the platform and I think I sent unit 1 and 2, if I'm not lying. I sent unit 1 and 2 to the platform. So I told you people that because I'm handling different centers, you people, I'm handling 7 centers, and I cannot ask each center to create a platform so that I will be sending you slides. So at least the course rep or the leaders in the platform can just create the platform, send the link to your colleagues, let them join. Because if as at now, 1 week now, you don't have the slides, then you have a problem as a student. And you can't complain that particular one when we have a class. My number is there. If you want the number, you can call. I think somebody chatted me last week or so, and I sent the person a particular number to call so that the person will be added to the group. So my sister, you need to make a little better effort. But if you stay in your comfort zone for you to be added to the platform or get the slides, it will be very difficult. That is what I can say.
Please try my best. I contacted the woman, the madam at the river. I told the madam that he should add me into the platform so that I can also get access to the slide and information needed for the course. But no information up to now.
I was expecting you to call more.
I added you.
To make a lot of effort.
You've not seen yourself on the platform. Any number that called or sent me a message has been added. Are you saying you are not on the platform?
And listen, when you are added to the platform,
what is my problem?
Hello. When you are added to the platform after the slide, you will ask for us to resend it again. So that should be something that you should put in mind, that when you are added to a particular platform, the previous messages, you will not get them unless you request for them. So if you have been added recently, you have to request that I will just added. Can you you send the slide so that I can also get a. If not, it will be a problem. Let me proceed. Today I have a lot. Today I'm going to utilize all the 4 hours. But I have a lot to teach today. If not, you will not be able to finish what you have to do. So when I'm moving a little bit faster, try and get followers. Forgive me. Is that okay? Okay, so let me proceed to what I want to teach today. I think is it Mavis or something? Can you put on some micro? If it's a champion, can you mute yourself? All right. So let me proceed. Today we are going to look at DNA replication. Replica. Replication also we got it. It's an English word which means replica. Replica means a copy. A copy. So most of the you see a lot of people wearing football jerseys across. The one that we wear them in Ghana, we call them the replica. Replica. So it is not the original one, but a copy of the original one. That is the idea that should come to mind. So by the end of today, we are going to look at how we synthesize proteins, explain the following concept: replication, gene expression, transcription, and translation. These are some of the things that we will know. I want to describe how knowledge DNA can be applied in food and processing industry. I know by now some of you are saying, we came to learn home economics. Why are you people bothering us with this DNA, RNA? Yes, they are very, very important when it comes to food. Right? Because we don't have a proper system of checking our food safety. We may not see it important, but the developed world are using this thing to detect adulteration, fraud, and GMO, genetically modified food, and the rest. So this thing that we are learning, don't just capture it in your mind that we are not biology students. Why should we are not doctors. Why should we be learning how we inherit a trait from our parents and the rest? We also have it's important in our field of study. So we will look at them. So you look at some of the definitions and how DNA formation is done, RNA and the types. I think we spoke about some of them last. So let's look at DNA replication. Before I even look at the slides, let me just explain it, use a scenario to talk about it so that probably we can appreciate it better. When we say replication, we are just trying to say that you are making an exact copy of something. Exact copy of something. That is replication. So let's say you have a paper that you want to probably, let's use those people who stake the lotto, for example. So when you go to the lotto people, when they take their booklets, they will put inside a carbon paper. And when they write the lotto, they will get a copy. They will get that one, we call it duplicate because probably it's just a due. They make it twice, duplicate of it. So when they give you the original one, or I don't know whether the original one that they give it out or the duplicate that they give it out, but at the end of the day, they have made a replica of the original one, so that when at the end of the day you win the lotto that you stake, when you come back, they will just look at the copy that they are also having to confirm whether what you are saying is true, or for them to know the number of people who have won the lotto in that particular day. So that is what duplication means. When it comes to issue of DNA, in biology, our cells undergoes what you call cell division. Our cells undergo what we call cell division. So let me cast your mind back at your interscience and that of your biology, whereby you did what we call mitosis and meiosis. Do you remember these things? Do you remember that you did some cell division like that, mitosis and meiosis? It was a very common term that we normally use when we make. I know some of you are not science students, but aside even the science, we did this meiosis and mitosis way back in integrated science. Not only that, when it comes to reproduction or division, we have three main. We have mitosis, meiosis, and binary fusion. With the binary fusion there with the eukaryotic cell, the amoeba, and the rest. What happens is, before a cell will divide, let's say you have one particular sex cell and it wants to divide to become two. The first thing that should happen is, when the cell division occurs, each of the two daughter cells should have the same amount of chromosomes, and the DNA that they contain should be equal. So, because it is one cell that is going to divide into two, the DNA or the genetic material should come together and copy each other, or there should be a duplicate or replicate of each of the genetic material. So let's say, I don't know how to put it. Let's say a particular cell wants to divide to have equal amount of DNA in it. One DNA is supposed to divide into two, or it's supposed to replicate itself. So let's say that DNA was having something like, let me use this code. We are having a particular DNA that have A gene, E. And this particular this particular cell want to divide into two. It means that then the A here should copy itself and get another A. Then the gene here should also copy itself to get another gene. Then the C should also copy itself to get another C. So that when it divides, each of the two daughter cells will have the same amount of DNA or chromosome as compared to the parent cells. So that is what we call cell division. The cell division I told you that there are three types. That is not where our interest is. There are two that undergo DNA replication. That is the mitosis and that of the binary fusion. So our head that we see it on our head, our fingernails, these are all tissues that contain a lot of cells, and these cells divide to make it grow. So you go to the salon, you go and shave, and about 2 weeks time, your hair has grown again. The cells in your hair keep on dividing so that it will promote growth. Anytime cell division occurs, growth occurs. That is why we were born as kids, and now we have grown up to this level. So our body constantly undergo what we call cell division. And anytime a particular cell divides, the amount of the genetic material that is found in the daughter cell is always equal to the genetic material that is found in the parent cell, with the exception of meiosis, whereby with the exception of meiosis, and meiosis mostly involve when a man and a woman sex cell come together to form a zygote. With that one, the woman come out with a pair of chromosomes, the man also come out with a pair of chromosomes. So that one is not, it doesn't undergo what you call cell replication. So I don't want to go much into that particular concept. But what I'm telling you is, when we talk of replication, it is just making an exact copy of a particular DNA when cell division want to occur. So let me come back to my slide and see what the slide says. It is a creation of a copy or duplicate. So that is what it should come to mind. Replication is commonly associated with DNA in biology, where DNA is copied prior to cell division. So anytime a cell wants to divide, there is a replication of your DNA. Your DNA should be replicated so that when we pick one of the cells, it will contain genetic information as the parent cell. So that is what we need to understand. We are saying that it is very essential for organism to grow, but when cell divide, it must replicate the DNA in its genome. Remember, we spoke about genome last week, whereby I made mention that the genome is the total genetic material that is found in every organism. So we said that anytime cell division will occur, there should be a replication of the DNA in its genome so that the two daughter cells will have the same genetic information as their parent cell. Then there are some structures that I will use it to explain better. We said that before DNA replication will occur, remember I told you last week that DNA is double-stranded. So before the replication occurs, the double-stranded should be separated to single-stranded, and it is this single strand that will be attached to appropriate base to form for the cell division to occur. So anytime, remember, let me see if so you see, so you see, where my case is, all the DNA, they are double-stranded. It means that they are having double edges, and the middle is connected by the nitrogenous base. The nitrogenous base, the one that we spoke about, the guanine, the cytosine, whatever. Those are the ones that are connected to each other within it. So if cell division want to occur, it's supposed to separate it. That's why we call it unwinding. Unwinding. It's supposed to be unwind, and it is done by what is it? An enzyme that we call helicase. And that is the enzyme that is involved in this particular formation. So let me go back to the slide that I was talking before. I'll come back here. Don't worry. So we are saying that the two strands are separated, and each strand complementary DNA sequences recreated by an enzyme called the DNA polymerase. So what happened is, let me come back to this particular slide again. So you see the last one. Let's use the second one. The unwinding is happening in the second one, the second picture where my case is. So you can see that a particular enzyme is trying to separate the two strands, because the DNA is double-stranded. So it is trying to separate the two strands into single strand. And when the two strand was separated, the third one is where the division has already occurred. So the third one, you can see that each of the strand has also been attached by a specific base. And when I talk of a specific base, what I'm saying is that last week we learned that anytime that a DNA is having a base of adenine, the complementary base is supposed to be thymine. Please remember this. I taught this thing last week. Anytime a DNA is having a base of guanine, the specific or the complementary base that should come and pair it to make it complete is cytosine. Do you remember this thing that we did? Anytime there is cytosine, yes, anytime there is cytosine, the complementary one is supposed to be guanine. And I think this is a few things that we did, with the exception of when we came to the RNA, whereby I said that in RNA, anytime you see that the T is always replaced by the uracil. So don't forget about this knowledge that we shared last week. So you can see that with the last picture, each of the separated strand has been complementary by their specific base. So you can see now we have two different DNA strands over here, and each of them is double-stranded. And when it happens like this, we have gotten two different DNA. And when it occurs like this, this is where cell division. So when it divides, this one and this one are the same. These two daughter cells are the same. They are the same, and the genetic material that they contain, you can compare it to the mother cell or the parent cell, which my Keza is using it to show. And people, are you following? I know this is something that is abstract, that I'm trying to put it in a context for you to get it. Are you following? Hello?
Yes, sir. Yes, sir.
Are you okay with the explanation? Today, I don't want to move fast so that you can get the concept. It's very, very important for us to understand this particular concept before we move on. So what you need to know is, anytime there's going to be a cell division, the genetic material are things that we inherit for us to know that I belong to this particular parent and I belong to this particular family. Those genetic material might be divided so that when cell division occurs, each of the daughter cells will have the same genetic material like the parent cells. So that is what the point is trying to make. So the picture here depicts how the the DNA replication occurs. So the process in which a particular DNA make a exact copy of it is what we call the replication. The replication, that is the process. So you can see that it's the creation of a copy of duplicate. A copy or duplicate, exact one. So that your daughter, that is the main reason why your daughter resembles you. And the main reason why your daughter might not resemble you, it might resemble your husband, your wife, or any other member within your family. And even though if they don't even resemble them physically, which we term is as phenotype, genotypically or genotype, they may resemble the genotype. That is why we take some of our blood samples, we test it with our children to know whether they come from us or they don't come from us. So DNA that they do it, if they do it well, I don't think there will be a mistake. Anytime they test for it, there should be 99% assurance that the child belongs to you. So let's make some progress. We said that as DNA polymerase can only extend. Let me talk about that before. So what I'm trying to say is the unwinding. When you say you are unwinding, something has been wound, you are trying to unwind, you are straightening it. So the unwinding is done by the enzyme called helicase. But it is the DNA polymerase that help the other DNA to get the complementary base. So let's say after this particular base or this particular DNA has unwound itself and separated from one another, each of the separated strands should be paired with another base or complement. We call it complementary base, and that particular work is done by DNA polymerase. So that is a specific thing that I want you to please pay attention to the enzyme that I'm mentioning there. What DNA polymerase is, what is its function, helicase, what is its function. You need to understand this thing because probably these are some of the things that they can easily be asked. So we said that in a way that the base on the old strand detects which base appear on the new strand, the cell end up with a perfect copy of the DNA. So anytime, let me try and come out with this and see if you people could be able to help me to come out with it. Let's say we are having a DNA and the base pair a DNA, and we want to make a exact copy of that particular DNA. So the DNA is having at the first end is having A. Please, can you get a piece of paper or pen and write anything that I mentioned so that I will get your response and know whether you are following what they are doing. So let's say.
Please, question before you go. Sir, please, can you go back to the unwinding slide? Sir, please, you said that when you are unwinding, an enzyme called helicase will help you to unwind.
Yes.
But when you are replicating, then another enzyme, is it the polymerase?
Okay, so this is how it is done. The unwind, let me say you are washing. When you wash your clothes, let me just come back to what we do at home. When you wash your clothes, sometimes you squeeze it to get off the water inside the clothes before you go to the dry line and dry it. Is that not so? When you do it, when you finish squeezing the clothes, when you are done, you always unwind it so that the clothes will come back to its normal position. Sometimes when the cloth is very heavy, especially when you have a bedspread, two people, one person will stand at the end, another person will stand at the end, then one person will be twisting it you'll be twisting them to opposite direction. When I'm moving right, the other person should move left. So when you are done, you try to unwind it back or you try to twist it back. And we are saying that particular unwind that happens inside the DNA is facilitated by a particular enzyme called helicase. That is the helicase. Its work is to help to unwind, because if it doesn't unwind, how are you going to separate them? I hope you are with me. So that particular enzyme is called helicase. After the unwind is finished, anytime you finish with the unwind, you have two different strands, because we said that DNA is double-stranded. So you have two different strands. And because you are going to divide it, each strand should have appropriate base, or what we call the complementary base. So the body will make up the complementary base to attach to each of the strand to make the two daughter cells double strand each. And that particular work is done by the complementary base that attach to it to the unwind strand of the DNA is done by DNA polymerase. The polymerase. That is what I'm trying to say. Is that okay?
Yes, sir.
Are you okay with that one? Okay. Then I think
Yes, sir.
we can make some progress here. So just put it in your mind that at this point there are two enzymes here. One of the enzymes help you to unwind the DNA to get a separate strand, and that enzyme is called the helicase. And another enzyme will help you to add the complementary base to each of the strand that you have gotten, a single strand you have gotten to make it a double strand. And that particular work is also done by the DNA polymerase. So these are the functions of those things that I want you to understand. So I was coming to give you a work. Do you all have your paper and your pen with you? So let's say I have a DNA, and there are two bases. No, there are two strands. Let me say. So the first strand is having A. We are talking about the nitrogenous base that is found inside those ones. So we have A, T, J, D, J, A, T. We have made mention of 1, 2, 3, 4, 5, 6, 7. We have made mention of 7 different nitrogenous base inside that particular DNA. Now, this particular DNA or this particular strand need to get a complementary strand that will come and pair with it to make it a complete DNA. So once I have A here, remember A is the adenine, T is thymine, G is guanine, C is cytosine, another G is here guanine, another A adenine, another T thymine. So my question is, I want you to open your microphone. I want to see whether you guys are following what we are doing. My question is, when I have A, what will be the complementary nitrogenous base that should pair with the A? I have another T here. I have C. I have G. I have C. So I want you to write all of them down so that when I finish, you'll be able to tell me. Then you link it to each other. So that when I mention A, T, G, C, G, A, T, you will tell me that the corresponding base that should pair with it is going to be this, and that. I want everybody to be able to do this so that the work is going to be very easy for us, because once we don't understand this particular concept, it will be very difficult for us to move on. Can someone try? Has anyone finished? Anyone who has finished can just raise up the hand and I'll let the person come out with the appropriate nucleotide. Yes, Cynthia.
Sir, please, the A, that is the adenine, is going to pair with C, that is the cytosine. And J is going to pair with C.
No, take your time. Take your time. I want you to follow what I gave you. I gave you A, T, J, C, J, A, T. So I want you to move in that direction. Did you get it? The first one was A, T, J, C, J, A, T. I want you to know the appropriate base that is going to give it to them. So the first one, yes, you said that A is going to pair with T. Then the second one is T. And the T is going to pair with what?
With J.
T is going to pair with what?
That is J.
I can't
The
hear
guana.
you well.
So the J, the guana.
T. No. Then there's something wrong. A is pairing with T. And then if you have T, what will the T pair with? Can someone also come in? I want you people to get this particular. I'm not asking you to memorize this, but I'm just trying to let you know how it works, so that when the cell division occur, or when there is a duplicate or replication
So
of
please,
DNA,
the T
the
will pair
appropriate
with
base can be
Jane.
added. The T will pair with what?
J.
Jane.
Jane.
No,
Yes.
no.
Sir.
It means you've forgotten what we did last week.
No, sir. Sir, please.
Yes,
Per what you gave to us. Per what you gave to us. The timing will always go with the guanine.
The timing.
The guanine.
And then
Yes.
the adenine will go with the cytosine.
You
Hello,
agree
sir.
with that?
Sir,
We don't agree.
the adenine will
The
go with
adenine with the timing.
The adenine always pair with the timing.
Sir, please
The
let me try.
timing. It pairs with the
Sir.
timing.
Good. The adenine. Put it in
The
mind. The
adenine pairs with the
adenine
timing.
with the timing.
Mhm.
So you see, I said that the first one that she said was right, because the first one
Yes.
was adenine, which is A. It is going to pair with T.
Yes.
Now the second
Yes.
one is, so the T will pair with what?
T. With A.
It will pair with A. Very good. It will just pair with A. That is the
And
response.
then G will pair with C.
J will
C
pair
will
with
pair with G.
And then C will pair with G.
And G will pair with
C
C.
will pair
A
with
will pair
T.
with C. T will pair with A.
Good. Actually, I hope
Sir,
you
I
are not
don't
coming to have
understand. So,
a
sir,
problem with
please, no. No, I'm not having a problem with you. It's just a question.
Okay.
Please, sir, per the structure that you gave us the last time, the DNA structure, you use the colors, and then with the links, you said that the guanine, which is the yellow, will always pairs with the timing, which is the green, and then
Do you
the
all agree that
admin.
that is what I said?
My sister, please, it's no.
Sir, the
It's
structure,
no.
the picture.
No, Esme, I'm coming.
Yes.
Me, this is what I remember. I told you before that as for DNA and those things issues, you can't mix them. When you mix one of them, that is where we say that mutation has occurred. These are some of the reasons why you see a child who is having Down syndrome, you see a child with this. If you don't get appropriate base, it is not going to work. How do you get it? Let me come in. I said that the adenine always move with timing. Then the guanine always move with cytosine.
Plus cytosine. Okay.
I hope you remember. That is what I said. And when we were doing the RNA, that is where I said that as for the RNA, instead of the timing, it will be replaced by uracil. That is the difference that exists between RNA structure and that of the DNA structure. But when we come here with the DNA, always A is paired with T, adenine and timing, then guanine and cytosine. So that is how the arrangement is done throughout the DNA strand. So the one I gave you A, T, gene, T, gene, A, T. So with the A, I think the first two, or the first three, we've gotten it right. And once we've gotten the first three right, the rest is not anything. I hope you understand. I hope you understand.
I'm following. Yes.
Esmin, I hope so Esmin, if you have included that the T will pair with J, then probably we need to decode that from your mind. Now, code this particular one. A is always pairing with T, which is adenine and thymine.
And timing. Yes, sir.
J is always pairing with C, that is guanine
Cytosine.
and cytosine.
Okay,
So
sir.
now, Esmin, tell me the base that you are going to add to the following. T,
Okay.
J, are you writing T, J,
TG.
A,
A.
A, C? Tell me the appropriate base or the appropriate complementary base you are going to add.
So you said TG A AC.
And
So 4. So is it 4?
there are 4.
AC. So sir, then it means so the T will go with the adenine.
Mhm.
Then the guanine will go with the cytosine.
Mhm.
Then the adenine will also go with the thymine.
Uh-huh.
And then the cytosine the C will go with the guanine.
Good, good, good. I watched that video
Okay.
yesterday. I said, clap for your hand, clap for your hand.
Thank you.
All right, so that is it. That is
Yeah,
how I
please,
want
I
you.
don't understand. No.
Who is talking? Mommy Deborah, are you the one?
Yes, sir.
Are you the one saying you don't understand?
Yes, because
Were you in class?
I went offline. That's why
Were you in
the
class
network.
last week?
Yes, sir.
You people should listen. Teaching this particular thing is very difficult. If you don't get a stable network and you go and come back and tell me you don't get it, I'll have problem with you. I think that the lecture is being recorded, and I think after the recording they will give it to you. Were you giving the last week one?
Yes,
Were
sir.
you giving the last week one? So this week too, I'm hoping that it will be given to you. Please, if you know you are staying in an area whereby network is a problem, when it is time for online class, try to position yourself wherever you are going to get it, because I have to talk plenty for, and these things are something that when you go offline and come back, you will not be able to appreciate it. I have to be sincere. So, Madam Deborah, what I'm trying to tell you is, do you want? No, let me continue. If I continue to, you may not be able to understand. So let me see what I can do. Madam Deborah, where were you when you went offline? Where were you? When I was talking about
So,
this slide, were you
so,
around?
so please, yes, I was around.
So which part that you don't understand? I want to be specific so that I can tackle your problem. Which portion don't you understand?
So, please, you mentioned some letters.
Were you in class last week?
Yes, please.
Do you remember we made mention of some letters that I call it coding? I made
Yes,
mention of
sir.
adenine, cytosine. I use colors to differentiate among them.
Yes, yes, the guanine, the cytosine, and adenine.
And those are the letters. It's not any name, 12 letters. Yeah, it's not diason. It's not a dream. These are the letters that we use. That we call it code. That is how proteins are being coded in our system. Is that okay? So I told you that. You see, I told you that before a cell division occurs, there should be a duplicate or replication of DNA. I hope that one you understand that point. So when that particular DNA, when there's a before division will occur, DNA is double stranded. And I said that they are supposed to unwind so that each of the strand will stand on its own. And when each of the stand, what is it? When each of the how am I even putting it? When each of the strands stand on its own, they are supposed to get an appropriate or complementary base to pair with it so that it will form a complete DNA. And that appropriate base is being done by an enzyme called DNA polymerase. But the unwind is done by the helicase, that is an enzyme. And once the helicase has unwind it and it has stand individually or it is standing on its own, the appropriate base that should be added will base on the single strand that is standing. So if you have a strand that is having what is it nitrogenous base, or it is having the coding system to be AT change, the appropriate base that you are supposed to go to add is TAC, because adenine is supposed to pair with thymine. Thymine is supposed to pair with adenine. Guanine is supposed to pair with cytosine. Cytosine is supposed to pair with guanine. And that is how it is being done. It's moving in that particular direction until you have the full DNA. So I just asked a question that when I give you this particular code, recently I gave A, T gene, C gene, A, T for someone to provide the complementary base. And I think most of your colleagues have been able to do that. Then I came back to Ethne, who was complaining about it, and I gave her an example, TGAC, and she has been able to tell me that the T is going to be paired with A, the G is going to pair with C, the A. So my interest here is not for you to memorize these particular letters. That when you are moving around, you'll be talking about cytosine, guanine, adenine, thymine. No, that is not my interest. My interest here is when you understand this concept, you know that when you understand this concept, you'll be able to know that. Please forgive me, my daughter is disturbing me. You'll be able to know that this particular base is going to have the complementary base of this particular DNA. Is that okay? Are you with me?
Yes, sir.
Is anyone having any challenge again? It's already 8:21. We've not gone anywhere. Today we have 3 slides, 3 different units that we are going to work on. Let me continue.
Yes, sir. Yeah.
You are okay. Okay, so we are saying that each of the new strand, each of the strand make a new partner by adding the appropriate nucleotide by the help of enzyme polymerase. I think I have made this point already. The result is that there are now new 2 double-stranded DNA molecules in the nucleus, and that is the picture that I show you. So this is the point is talking about this. The point is talking about it. Now you can see that there is new 2, 1, 2 in the nucleus. Then so we said that so that when the cell divides, each nucleus contains identical DNA created through the process called replication. So what you are saying that the new 2 daughter cells are found in the DNA. So when the cell division occurs, one of them will join one particular strand or one particular cell, and another one will join one particular cell. So when you pick those two cells, the one that we call them the daughter cell, they have an identical genetic material. The information that is found here is supposed to be the same information that is found in the other cell. I think here we all understand it, so we can proceed. So this picture also depicts the same thing that I was explaining. So you can see that we have a new strand here. This one, old strand and old strand. You see the last ones are the old strand, and the middle ones are the new strand. So the old one, we just made a copy here. So you can see the colors. Then we get appropriate base to pair with it. So you can see the first one. Let's look at the old. The first one was color green, and it was pairing with color yellow. So you can see that when we separated them and we are adding a new strand to it, the new strand also came with color yellow. Can you see that? Where my kids are? Can you appreciate that? Can you appreciate that?
Yes, sir.
All right. Then you can see that the second one was the old strand was color. Is it orange or red? Me, my eyes, I can't see proper.
Orange.
And
Orange.
the old strand was somehow blue, right? The sea blue. So you can see that when the unwind occurs and they are trying to add the new strand or the new base, you also have the same color that's supposed to be added. So that when you don't add the same thing and it changes, then you can see cell mutation occur, or the kind of protein that is going to be formed will also change in shape, and its basic constituent, the thing that it contains. So this particular one is there for you to understand. So let me proceed. Now let's look at protein synthesis. Now we've looked at DNA. We've talked about how DNA unwind itself and the stuff. Now we are coming to talk about protein synthesis. Please, I want you to remember that when we talk of protein synthesis, someone will say that how does we synthesize protein? Protein is something we find in our food and we eat. No, when you take in your food, your body doesn't see what you have taken as protein. Your body rather see amino acids. That is why we said that the end product of protein is called amino acids. So your body doesn't see that protein, but rather your body rather uses these amino acids to form a protein. So a chain of amino acids come together to form a protein. Esme, your hand is up, or it was the old one?
Sorry.
All right. So different type of amino acids come together to form protein. And let me talk about it. In our system, most of the things in our systems are made up of proteins. We can talk of enzymes, we can talk of hormones, we can talk of certain structures in our body. All these things are made up of proteins. So if the body is not able to synthesize the correct protein, it is going to be very difficult for the body to function properly. So, and it is these proteins that help our cells to grow. It helps our cells to divide. Because through the cell division, it promotes growth. So when you were young, you were very short. Through cell division, your body has been, the cells have been divided, and now your hand is longer than when you were a child. It is through cell division that that particular process has occurred. So I just want you to have that notion before we proceed. So in protein synthesis, it starts with what we call gene expression. Gene expression. And we say that in gene expression, it's gene expression, we say that it is a process by which information from gene is used to synthesize functional gene or product, or functional gene product. So in gene expression, the process whereby how gene is used to effect an organism trait. So let's say my body want to synthesize lactase. Let me put it. Lactase is an enzyme that digests lactose, and lactose is sugar that is found in milk. The milk sugar is called lactase. If you remember this thing, we have lactic acid. That is the acid that is found in milk. So lactase is the sugar that is found in milk. So now, as a child, when you are born, you have been breastfed, and this particular lactose is found inside the milk that has been given to you. So your body needs to synthesize a particular protein enzyme that is going to break down the lactose into simple units for the body to use it. And that enzyme that does that is lactase. So the lactase is made up of protein. So your body should find a way to synthesize that particular protein. So before that particular protein can be synthesized, there should be a gene expression. There should be a gene that will code for that particular lactase that is going to be synthesized, so that the body will know that, because each of the product or each of the protein product in the system code for something different. When you have a T gene, it is going to code for specific structure or specific product. When you change the C and make it gene, probably you will not get the specific product, or when that particular protein is being produced, it cannot perform the same function as the protein that you were trying to synthesize. So I want you to put it in your mind. Anytime your body wants to synthesize specific protein for something, there should be a particular gene that will express, that will show that this is the kind of gene, or this is the kind of product that I want to synthesize. And it is done in a coding manner. And all these things, it involves the cytosine, the guanine, and whatever that I've been talking about. And most of the time, it moves in three. Each amino acid pair with three. It is three codon. We call it codon. Three codon that pair for each amino acid. So there should be a gene that will express. When the gene is expressed, when lactase gene is expressed, it will tell the body that the body wants to synthesize lactase, so that it will combine appropriate amino acids to come together to form that particular lactase. And they are very long. Sometimes you may get one particular structural gene, a protein that have about 150 different amino acids that has been linked together. I think when you have time, look at insulin, look at hemoglobin A. You know, hemoglobin, we have different types of hemoglobin A. When you look at all those ones, they contain what we call the alpha and the beta chain. And the chain are very long. And we cannot go into that. One will be the work for the doctors and those people in the lab to deal with. But what I want you to understand is, anytime your body wants to synthesize specific thing, a protein structure, there should be a gene that will express for that particular structure. And when that gene is expressed, the body will understand that this is the kind of protein that the body wants to synthesize. And we say that this product are often protein, and the gene that code for amino acid sequence are known as the structural gene. So the gene that will code for specific amino acid sequence or amino acids is called the structural gene, And as I say, there are other coding genes that are not used in protein synthesis, but in synthesizing of functional RNA. Remember, when it comes to the synthesis of protein, it involves specific RNA. You have the messenger RNA, transfer RNA, and the ribosomal RNA. Remember, the transfer RNA, its work is to transport the information that the messenger RNA has moved from the nucleus into the cytoplasm. So there are other coding genes that are not involved in protein synthesis. Some of them are into metabolism. So metabolism, some of them are involved in the breakdown of glucose into pyruvate or ATP for your body to use it to generate energy. So that is what, and those ones, we call them that non-protein coding gene. They are non-protein coding gene. They are not coding for protein synthesis. We are saying that the process of gene expression involves two main stages. So we are going to look at the stages that are involved in gene expression. There are two. We have the transcription, then we have the translation. We have the transcription, then we have what we call the translation. So let me talk about them. Let me quickly talk about the translation and the transcription. So let's talk about the transcription. So the transcription, when the body wants to synthesize proteins, it involves three main RNA. There are three main RNA that are involved. There are a lot of RNA in our system. There are not only three, but in our context, we are looking at protein synthesis. With the protein synthesis, only three RNA that are involved. There are some that we call the micronucleus RNA. There are a lot. So those ones are not important to us. So we say that the transcription, in the first stage of protein synthesis, where single-stranded messenger RNA is copied from DNA code strand. So let me put it in this way. We know how DNA is. We look at how DNA and how it is. So messenger RNA is always copied from the DNA. And remember, I told you that the DNA is always found in the nucleus. So the messenger RNA, because protein synthesis does not occur in the nucleus, rather it occurs in the cytoplasm inside the ribosome. There should be a coding information that should be sent to the ribosome, so that the ribosome will base on it to manufacture the specific protein that has been sent to it. So the messenger RNA, remember last week I told you that the messenger RNA, its function is to carry genetic information or protein coding information from the nucleus into that of the cytoplasm. Then the transfer RNA will just carry that particular information into the ribosome for the ribosomal RNA to do the protein synthesis. So the messenger RNA is a single-stranded. Remember, I told you that RNA is single-stranded, and we copy it from DNA. So let me create an example here again. Now I have a DNA. Let me say I'm taking only one strand of DNA. I have a DNA which is please, the code that I'll be mentioning, I want you to mention them. I want you to write them so that you understand. So let's say I have a DNA of J T A C T G. I have a DNA strand of J T A C T G. That is one strand of DNA. And I want to copy messenger RNA from it. I want to copy an RNA from this particular one. Remember, there's one thing that I want to, this thing, if you want to copy messenger RNA from here, you are going to just give it an appropriate complementary base as we did when we were doing the DNA replication. By here, the only difference is, remember last week I told you that in DNA, in RNA, anytime you see timing, you replace it by what? Who remember?
Uracil.
You replace it by what?
Uracil.
Good, uracil, which is U. So when I look at this coding figure that I've given to you, we have J, and J is 19. So J will pair with what? J is going to pair with what?
Cytosine.
Cytosine. J is going to pair with what? Cytosine. Good. Then we have timing here. And we say that when you are dealing with issue of what is it? When it comes to issue of RNA, timing is going to replace by what?
Uracil.
Uracil. So the timing at the normal circumstance must pair with what?
Adenine.
Adenine. Good. So the T will pair with what? A.
A.
Is that also?
Yes, sir.
And the A will pair with what?
A. Sir, A will pair with T. The uracil.
But we say that in RNA we don't have T, we have rather U, uracil. Is that also?
Yes.
So with the A, I'll write my U here.
Uracil.
Then the T will pair with what?
Zoom second one face to face.
So the T is adenine. The C
will pair with what?
The G will pair with cytosine.
Who is speaking her language here?
So that's
Sister Haku.
You come and see the way I'm forcing for you to understand it, and you are speaking your hometown language. The come group guys are already have it. You know where I come from. I'm from Burkina. Okay. So the C is going to pair with the guanine, with the G.
Yes.
Then the T will pair with what?
Adenine.
Adenine. Then the G will pair with what?
Cytosine.
The cytosine.
Good. So you can see that this new strand that we have created here is what we call the messenger RNA. So this particular messenger RNA that we have created from the DNA will now move from the nucleus into the cytoplasm. Into the cytoplasm of the cell. And we know cytoplasm contains a lot of organelles. Most of the organelles are within the cytoplasm. So when you get to the cytoplasm, then another RNA, which is called the transfer RNA, will now pick this particular code and send it into the ribosome. Then the ribosomal RNA will now work on it to let it form different type of amino acids. So what happens is, anytime the transfer RNA is transferring this particular coding into the ribosome, it moves them in three. Each amino acid has 3 codes. Each amino acid have 3 codes. Each of them. So when you pick, I told you, I think I've been using valine. Valine is having 3 codes. It is CGA. So anytime the transfer RNA CGA, it will pick it and send it to the to the ribosome. So anytime when ribosome see CGA, automatically you know it is going to code for valine. Then the valine will be produced as an amino acid. Then it will continue. Probably another one will come with methionine, which is another amino acid. Other one will come with glutamic acid, which is another amino acid. Other one will come with leucine, isoleucine, tryptophan. All these ones are amino acid, and their base is only 333 code. Hey, I'm confusing conformer.
Yeah,
Are you following?
Say do it.
No, I was confusing it.
Confusing, yeah. Don't worry, you understand.
Yes.
You understand. Biochemistry is not always easy for you to get it, but you will get it. The way I've taken my time teaching you this concept, if you decide not to understand it, no one should. Pay attention to what I'm saying. And I want you to write the things that I asked you to write. Because if you don't write, I don't have board in front of me. If I'm teaching this thing face to face, it would have been easy for me to illustrate them on the board. But because it's an online, you just have to appreciate it. Hey, where is my friend Ula? Where is Ula? Ask me, where is Ula?
She came.
What did this?
Sam said we fiso. The line not dropping. No was one paying.
So all right, so let me continue. So I just use this particular one to explain the whole process. The whole process of transcription to translation. I'm actually, you know, I finished teaching transcription and translation together. So let me go back to my slide. So we said that in the first stage of protein synthesis, where a single-stranded messenger RNA is copied, messenger RNA copy is made from DNA coding strand, and that is what we have done here. So the DNA was the one that is JTACTG. Then we've been able to copy messenger RNA from it, and the messenger RNA we have CAUGAC. I hope you all understand this point. We are saying that the enzyme that also help us to produce this thing is called RNA polymerase. Do you remember we have met DNA polymerase somewhere? Do you remember we met DNA polymerase somewhere? Do you remember we met DNA polymerase somewhere, my people?
So you mentioned
Yes, sir.
Yes, and what was the function of the DNA polymerase? What was the function?
So it does the complementary base. Is the complementary base.
It is not a complementary base.
So it helps to get different strands.
Yeah, it helps to add the complementary base. It helps to add, put it in that concept or that perspective. It helps to add the complementary base to the DNA strand. It helps to add the complementary base to the DNA strand. Because if you write it, it is the complementary base of the DNA strand. Probably, so we also have what we call the RNA polymerase, and its work is that it helps in the production of messenger RNA. So the messenger RNA that we copy it from the DNA, the enzyme that facilitates that process is called the RNA polymerase. And this process is what we call transcription. Transcription, you are transcribing a DNA into RNA, and through that, certain bases are changed, and one of the bases, specifically that changes, the timing always replaced with urea cell. I think that particular concept all of you understand. And we say that the genetic information in the DNA is copied into messenger RNA. The genetic information is what we have copied here. The coding numbers. So each of them contains specific genetic information. We say that the RNA is not directly involved in the protein synthesis but serve as a temporal copy of information found in the DNA. Please remember this point very well. They are saying that the messenger RNA, what we are saying is it does not involve itself in the protein synthesis, but rather it carries information that is used to synthesize protein rather. So it is not the messenger RNA that is going to synthesize the protein, but rather its information will be used to synthesize the protein. So that is what you know, because the messenger RNA is just sending the information that hey, the body needs to provide to synthesize probably insulin, and the insulin needs valine, methionine, this, and so the body should look at the code in the protein. So that is the point. And we said that the process of transcribing DNA into RNA requires change of information from double-stranded to single-stranded. We did this thing last week, and also timing it to uracil. We said that. So have you seen that with the issue of messenger RNA, the DNA itself it is not double-stranded as compared to the DNA. RNA is not double-stranded, it's single-stranded. So the process requires that it changes from double-stranded to single-stranded molecule, and also the timing in the base will also be changed into uracil. And I think I've made mention of this thing too. I think some of these things, some of them will surely come in an exam. Some of them will surely come. That one day I can assure you. I'm not saying I'm superhuman and I know the question will come, but most of the time when you are the lecturer and they are teaching, look at where the stress is most, and that is where most of the time you can just get some of your. And when you listen to my lecture, you know most of the places that I give a lot of stress to it. You should be able to understand that those ones are very, very important. So these are some of the coding and genetic coding that is done. So look at the picture on your screen. So we said amino acids and protein formation. So we have cytosine, adenine, and adenine. So you see valine is CAA. CAA, code for valine. Then we have CGA. CGA code for alanine. CGA code for alanine. Then you can see the one down here. So we have CGA, alanine, CAA, valine. We have CCA. Is it glycine or what? Then we have CGCT, adenine. Then we have gene, proline. Gene is proline. Then we have gene, which is leucine. Then we have CCA, which is glycine. So these are how proteins are being coded. This is how proteins are being coded. So let's see. So we said that a group of 3 bases control the production of amino acids in the cytoplasm. Each 3 consecutive base, that is, we call it codon. So you see these 3 bases here, these are what you call codons. They contain the code to produce specific amino acids. So when you link all these amino acids together, then you have gotten what you call protein. A particular protein has been synthesized. Sometimes the protein can be 3 or 4 amino acid chain, depending on the kind of function that is going to perform. Is that okay? Please, are you with me? Today, nobody can say I'm moving fast. Nobody can say that. Me, I can say that today, I'm moving slower than slow. Are you with me? Anita will say, Gloria.
So we are with you. We are with you.
So we are with you.
I hope it is moving on well.
Yes.
But I said the other 3, no, we didn't hear you.
The other one.
The 3 codings, and we have the vena and then the adenine, but the other 3, no.
The other 3, like I didn't get it.
So the codings, you know. I think they were 4, yeah, 3, 3, 4.
334.
Mm.
Not getting what you are saying.
Sir, please, you said each codings have 3. They move in 3s.
Yes, each 3 code, each 3 of the nitrogenous base will code for a specific amino acid.
Yes, please.
And that is the example that they have given, that if you have cytosine, adenine, and adenine, it will code
And
for
that
venom.
is venom. Yes, please.
If you have cytosine, guanine, and adenine, that one will code for alanine.
Alanine.
You know there are others down here that will
Okay.
make it very short. CGA, alanine, CCA, CAA, valine. Then you can see we have gin. So you see, we can have gin. GGG will code for proline, which is another form of protein. We have GAG, which is leucine. So please don't go and memorize this thing. Don't go and memorize this thing. They are not important. Yes, that's important for you to appreciate it, because nobody will ask you in the exam that what specific code or codon code for glycine or adenine. No. We will not be that weak.
Okay.
We will not be that weak. And I'm going to probably a question around that. So let me continue. All right. Please, I just want you to follow it step by step. You understand? It is something new. So when you listen very carefully, you jot certain things down, you get the slide and read them. Probably you appreciate them. I'm not expecting you to get everything here. That one day, this one is not magic.
Yes, sir.
All right. So remember, I spoke about the code.
Yes, sir.
Remember, I was talking about the codon, and I told you that the codons are the specific, the three
amino acid
for specific protein. So we say that codon is a sequence of 3 DNA or RNA nucleotide that correspond with the specific amino acids. So when you go back here, the codons are the numbers, and each 3 number correspond with a specific amino acids. Specific amino acid, each 3 codons, and it moves in 3. So that is how it is done. And we are saying that DNA and RNA molecules are written in language of 4 nucleotides. Meanwhile, the language of protein includes 20 amino acids. So remember, the DNA and RNA, they are always in 4 codons. If you remember what you were doing, the codons are, we have the thymine, we have the adenine, we have the guanine, then we have the cytosine. They are 4. But if you want to code for specific amino acid, you use it only 3. We don't use them. I hope you are with me. We use it 3 instead of 4. But in all, they are 4. And even in RNA, it's 4. In DNA, it's 4. In RNA, in DNA, it is TAGUJC. But in RNA, it is going to be UAJC. I hope you understand.
4.
They are 4. The DNA is TAGC and the RNA is UAGC, because we said that the timing is going to be replaced by the uracil. Do you remember that? So that is what that is what the point is trying to. And we are saying that these 4 languages can be used to produce or can be used to code all the 20 amino acids. Now, amino acids are many. They are many. They are not only 20. But we are looking at 20. There are a lot of amino acids that have been identified. But the specific one that we see them to be of importance is the 20 amino acids. The 20 amino acids. So we can use only these 4 code to code all the 20 amino acids. And if you have time and you go online, then you type the structure of all the 20 amino acids. The code, you will see that these are how the coding uses only the 4. Only the 4 that we use for the coding. So that is what we need to understand. And we have what we call the start and the stop codon. So we said that the start codon marks the site at which the translation into protein sequence begins, and the stop codon marks the site at which the translation ends. So anytime you are going to synthesize specific amino acids or protein, there should be a place that it will start. Then we have the stop codon. So we said that there are 3 RNA stop codons. We have the UAG, UAA, and UGA. So you see this one because they are they are RNA. You can't see any T here. You see, because they are they are RNA. There's no T. If immediately you are talking about RNA, you see timing inside, automatically you are wrong. So these are some of the few things you can pick it from there. They will say that when a triplet-based code for different amino acids, they have to be assembled to make a protein. So when you have a triplet base, so we say that each amino acid have a triplet base. Each amino acid have. I think that is the structure I should do earlier. And we say that when they are done, when they code for different amino acids, they have to come together to form a protein. And remember, amino acids are proteins are a group of amino acids that have been linked together, either by peptide bond or hydrogen bonds. So depending on the sequence in which it is being used. Then we can also say that the sequence of triplet code in the DNA molecule may code for a complete protein. Yes, so a particular triplet code that is picked from DNA can code for a complete protein, because if I have, let's say if I have DNA, which is gene C, T, and probably the T is replaced by U from the DNA, and this one gets to the cytoplasm. So with the gene CU, I can get gene, which is proline, which is an amino acid. I can get gene CC, which is another amino acid. I can get CCU, which is another amino acid.
Okay.
I hope you are with me.
Yes, sir.
So
Yes, sir.
only three base.
Yes, sir. Yes, sir.
Only three triplets. What is code that has been picked from the DNA can be code for a complete amino acid. And a complete amino acid, you can get about a complete protein. You can get about 3 or 4 of amino acid that has been linked together. So that is what the point is. And they say that there may be thousands or more bases in one gene. The different amino acids and order in which they are joined up determine the sort of protein being produced. So the kind of protein that is going to be produced determine the kind of amino acid that they are going to use. So each protein have, I think yesterday, let me see if I wrote something. I'm coming. So let's say, no, I didn't write them. So let's say you have something like, yeah, I wrote something here. Hemoglobin A. You know hemoglobin, the one that is responsible for carrying oxygen throughout your body, and the hemoglobin, and it is responsible for the red color of your blood. So it is a protein component, and that one too, the body code it. So we have hemoglobin is made up of 2 different sets of amino acid chain. We have the alpha chain and the beta chain. So when you come to the alpha chain, we have the valine, we have the leucine, we have the proline, we have the alanine, we have aspartic acid. So you see, when the body code for all these things and they are joined together, they will come and form what you call hemoglobin, and they will start carrying oxygen within your body. If you take one of the amino acids, or let's say I take up the proline, it means that it will not be able to perform its function. Because remember, in our basic biology, we were told that proteins perform its function based on the structure, based on the size, based on the physical structure. So if you alter the structure, or if you change the structure, you have changed the function of the protein completely. So that is something that we have to also understand. So this is the transcription and the translation. So you can see that under the transcription, messenger RNA will be gotten from. The first one is the DNA, so messenger RNA will be copied. So you can see the second one, RNA has been copied. So when you are copying RNA from DNA, we call that one as transcription. And when the body uses the code from the RNA to produce protein, to produce protein, then probably we say that you have done what you call translation. Because the translation is where the body will be able to use the gene to know that the gene means proline. That means that you have translated the code into a specific amino acid that you are looking for. So all these things that I've explained, these are the basic diagram you can use it to understand. So DNA, from DNA you get RNA, and this RNA, they are, remember the RNA, we have the messenger RNA, the transfer RNA, the ribosomal RNA. Then probably we have protein. Please, are you with me? Don't run away, leave me
Yeah,
here.
when you use the DNA to copy the RNA, it's called what?
Yes, sir.
When you use the DNA to copy the RNA, don't put it that way. Put it in this way. When RNA is copied from DNA,
Okay, yes, sir.
it is called transcription.
Yes, sir.
It's called transcription.
RNA.
Yes, and
RNA
when the
is copied.
so, and when you are able to synthesize protein from the what is it, the information that was carried by the RNA,
Yes, sir.
we call that translation.
Sir, it's called.
I said that. Listen, let me come again. I said that, you know, the structure that I showed you first. I said that we copied RNA from DNA, and that process is called transcription.
Okay.
You are copying RNA from DNA. It is called transcription. And the information that the RNA has copied from the DNA, we use it to synthesize amino acids and proteins. And when we are using that information to synthesize protein, we call it translation.
Okay.
So it's like someone speaking an Ewe. Me, I don't understand Ewe. And you are able to translate the Ewe language into English for me to understand. You have translated the language. The information that the person sent you, you've been able to translate it to me so that I can also understand. Are you with me?
Yes, sir.
Hello, sir. Please.
Hello.
Hello, sir. Please, I didn't get it last time you talked about the translation. I didn't get the
I'm not giving this definition here.
Yeah, please, I didn't get it.
You talk a salad alone. You're asking me definition. I didn't give definition here. What I'm saying is that if your body wants to produce protein, it goes through certain processes. The first, the thing that it starts with is gene expression. And I told you that Vivian, be careful.
I'm sorry.
So we say that it goes through certain processes. And the first thing, what it does is we call it gene expression. Remember, I spoke about gene expression, whereby specific gene will express and to show you the kind of protein that it wants to synthesize. Then we said that under the gene expression, we have two main types, two main processes that occur. We have transcription and translation. And I said that the transcription is a process whereby RNA is copied from DNA. And before RNA is copied from DNA, there should be some form of changes. Remember, I said that with the DNA, they are double-stranded, but RNA is single-stranded. So that information will change. Then also, in the DNA, the coding material contains thymine, but with the RNA, it is only uracil instead of thymine. So when these processes are done and RNA is copied from the DNA, we said that process is called transcription. The process is called transcription. Then after transcription has occurred, we have now produced what we call RNA, and specifically messenger RNA. Then there will be another process that will translate this, because messenger RNA is just carrying an information. So when that information is being translated into protein synthesis, then we say that we have done what we call translation. So let's say somebody stole your item, the person has run away from probably Accra to Kumasi. Then you go to police station, they will give you a letter that send it to Swami police station in Kumasi. When the Swami people see the letter, that particular letter is, I can say the letter is called the RNA or the messenger RNA. They will read the information on the letter and go and effect an arrest. They will go and arrest the person. When they're able to arrest the person, it means that they have translated the language on the letter to go and arrest the person. So that is what we call translation. So when you are copying proteins, or when proteins are being synthesized from the information that the body gets it from the messenger RNA, we say that translation has also occurred. Amatu, are you okay now? Are you able to
Yes,
me?
please.
I will
Thank you.
explain further. Without the means, I won't talk again.
Sir, please, I don't know if at the end of the lecture I can get your number,
No, this is a
please.
question for. All right, no problem. Oh, my
No.
number is even on the slides. When I put the slide on the platform, you see my number. Anyone who want to talk to me, you have
Sir,
to.
please, I'm not on the
Anyone who
platform.
wants to talk to me, you have to see Esme first. Esme is my personal secretary. So
Yes,
let's
sir, I'm here. Yes, sir.
let me continue. All right. So in the protein synthesis,
Thank you.
there is a flow of genetic material from one particular end to the other, and the process, the system in which, or the process in which all these particular processes occur, we call it the central dogma. The central dogma. So the central dogma, we are not just coming to explain anything different. We said that it describes the flow of genetic information in the cell from DNA to messenger RNA and protein. So the process whereby transcription occurs, translation occurs before protein synthesis, all these processes called or is known as central dogma. And we say that it states the gene specifying the sequence of messenger RNA molecule in which it turns specify the sequence of proteins. And the DNA, so with the central dogma, the gene expression will occur. When a gene is being expressed, a specific gene will express, oh, a gene has been expressed like, let me code for, let me code for, let me say insulin. Let me code for hemoglobin. That is the gene expression. A particular gene will express. Then probably that particular information will be used to produce the messenger RNA. Then the messenger RNA in the process will also include what you call protein. Protein itself, or amino acids and protein. So this is a few things that I want you to add so that you'll be able to understand. Say that the coded genetic information hardwire into DNA transcribed into individual transportable caskets composed of messenger RNA. So most of the information, the individual cassette. So it's like it is being moved in the form of compact bits, so that when they come out, the body will be able to read. So let's see, when you have a DVD, you know, at first we were using some old cassettes that have some wires. Those of you that you were born in, I know all of you are senior citizens, so I know you know what I'm saying. The old cassette that we were using. So the cassette were having some kind of code on it. So when you put it in your machine will be able to detect the code and display it for you. So you see, there are a lot of things that we use DNA, and that that experiment of DNA can even be used in different techniques. All right, so this is the picture of the central dogma. So the whole process, the gene expression, from DNA to RNA, from RNA to protein synthesis. So this is the whole process. And the same picture is here. So you can see the DNA double-stranded. So transcription from DNA to RNA, then from RNA to protein. Okay. So with that, from the RNA, you know, the protein here is made up of individual amino acids. So individual amino acids will come together to form the protein. So here is the question. I know this question too has been going through your mind. Obi Misada, hey, Nasser, what am I using this RNA for as a home economics student? Have you been asking yourself?
Yes, sir.
Maybe some
Yes, sir. Near the ending. Aduane na menko no.
Yes, sir. Yes, sir.
Yes, sir.
So I want you to understand that you are not only being taught to go into the kitchen and go and cook. You are not a catering student. Put it that one in your mind that you are not a catering student. You are home economics student. Some of you will go up and become dietitians, nutritionists, food technologies, and stuff. Food biochemists. So this course will even help you to understand the reason why certain processes are done to certain food. So that is what we are going to look at now. How are you going to apply this particular thing that we are learning to that of our food industries? So there are things that we need to know when it comes to this particular thing. So the first one is A and T or AT is a double bond and they are weaker, while CG is single bond and they are stronger. So you see, some of the food, because food products are all living organisms, they all have DNA in them. And it's the DNA that makes the physical appearance that you see. Some of the DNA will make a particular food look this hard. Some of them will make it look soft. So it is the genotype, genetically makeup of that particular food that you see it physically. So remember last week I told you that your physical appearance is based on your genes. So if you see me, I'm very dark, it means that I have a darker gene inside my system, or I inherit a darker gene from my parents. So that is what we are doing. We are saying that when you have adenine, food that contains adenine and thymine, that particular food is having a double bond. Hey, let's talk about this double bond here. Let me talk about it because we will talk about it when we are doing when we are talking about fat and oil. We will look at those double bond and single bond. Anytime you have any food that has a double bond in it, that particular food is weak. And when a particular food substance is weak, it means that a little bit of heat will break it apart. So that is one thing that you need to know. So when you have most of the soft food that we have in our system, they contain A to T. So something like banana, banana is very soft. When it is ripe, it is very soft. So it means that banana is made up of adenine and thymine, and the bond is very weak. The bond is weak because I think it contains two hydrogen bonds. The bond is made up of two hydrogen. Then any food that contains C and G contains single bond. And single bonds are very strong because they are stable. All the food that are single bonded, they are stable. Double bond, they are not stable. They are always reacting with the environment that they find themselves. So you can talk of something like food like yam. You need to cook yam for a long period of time before it softens, as compared to banana, whereby when it is down, it can easily become soft by itself. So you need to know that. Then we are also saying that A and T strand-rich food would require less heat than T and G-rich food. Who can tell me this? I think I have made mention of it in my previous explanation. That when you have food that contains A and T strands, the food will require less heat than C and G-rich food. Who can tell me? Who can tell me? I think I have spoken about it. Yes, now. If you were listening to me, you should be able to tell. Who wants to talk?
Sir, please, can I try?
They are soft.
Right, the one more person now can say or if you want to talk, if you cannot raise your hand, just draw my attention before you talk. Yes, who want to talk?
Sir, please, can I?
Yes, Vivian, yes.
Sir, because they are very soft. They don't require much heat.
Yes. When you are cooking something that is very soft, you don't require much heat. So let's see tomatoes. When you are cooking tomatoes and that of most of the time when we are cooking certain food in the house, when I'm doing a PC and I'm using tomatoes and garden eggs for stew or for whatever I'm using it for, I'll put everything together and start cooking them. Right, to get to a point, I have to just open the lid and remove the tomatoes and the garden eggs. Is that not what we do at home? So we remove them.
So either you do it like that or you allow some of them to boil at some point, then you add them.
Yes. So you see, it is telling you that some of the food, the one that it will get cooked easy, require less heat. So when you cook it for about 10 minutes, it is cooked. It becomes very soft. Then you remove them. So those one require less heat, and it requires less heat because of the A and T strand that is found in it. Those ones, they are very weak. They are very weak bonds. And those bonds, I said that the weak ones contain 2 hydrogen bonds, and the stronger ones contain 3 hydrogen bonds. All right, so that is what I can say about it. So these foods, most of the time, the cooking period is shortening as compared to, so A and T foods, their cooking period is shorter as compared to C and G. So it means that when you are cooking food that contains C and G, you have to cook it for a long period of time before that food will be cooked. Example would be what? Willy, and beans. Cooked. You see, those foods, you can cook it for, some of you have to add certain things to make it soft at a point in time. All right. So that is what I can say when it comes. So you see that it is important in our industries. Is it not important? Is it not important? So
Yes,
it's important. At least it will tell you that because of this particular, I need to break the bonds. The more the bond is higher, the strong, the more it requires a lot of energy. When the bond is very strong, it requires a lot of energy to break those bonds. But when the bonds are weak, little energy will cook it. Little energy will just cook it. Now, DNA is also a useful instrument in assessing food quality. It's very, very true. DNA, they are very useful. Yes, Esme. Esme, I can see your hand up.
Yes, sir. Sir, please, a question. Sir, talking about some foods being A to T, and then some being C to G. Please, does it mean that there are some foods that initially they are C to G, but as time goes on, without heat, they change to become A to T?
Very good, very good. So what you need to understand is this DNA, this RNA, and what most of them, we also have environmental issues. Environmental issue that also affects them. I hope you get it. So something like when banana is not ripe, probably the bond within the banana will be very strong. The bond will be very strong because it will be more harder. But the ripening process is also facilitated by certain enzymes and environmental temperature. How we get it? So by the time it will become ripe, its actual or its last, or let me say the A and T in it will express itself better than the C and G in it. That is what I can say. Basically, we can also, let me link it to humans. We have something that we call obesogenic gene. Obese. There are some families that when you go, everybody is fat. Have you noticed that thing before? There are specific families that when you go, a lot of the family members are fat. In biology or in nutrition terms, we call that particular gene, the gene that causes that particular thing, we call it obesogenic gene. But because once you inherit that particular gene from your parent, doesn't mean that automatically you are also going to be fat. There are certain environmental factors that you can do to suppress that particular gene. You can do that to suppress that particular gene. So let's say I inherited obesogenic gene from my parent, and this obesogenic gene are responsible for for what we call the adipose tissue. Adipose tissues are the tissue that stores fat in your system. But you can restrict yourself. You can decide to eat certain type of food that will suppress this particular obesogenic gene to make it less functional. So in the process of food, there are certain food that initial stage, when they are being matured, they may contain C and G, but the environmental system will not make that particular one active at a point in time. So let's say you pick banana. At first, banana is very hard. Then you put it in an enclosed area whereby you give it a special temperature. Then the temperature will make it very soft. So it means that you have suppressed the C and G in it, but you have made the A and T in it very active. Are you with me? Esme, are you with me?
Yes, sir.
Very good. So that is how it is. So there may be C and G component in it at the initial stage, but when it gets ripened, that particular C and G will not be active. It will be suppressed. Then the A and T will become very active at that moment due to environmental change and the enzyme that work on it. And remember, with protein, there are certain enzymes that we can use to change the structure of protein and the rest. Probably that will not be your field. Let me not go there before you start running away. We started with 42 people, now we are 39. So I can see people are running small. Okay. So we are saying that DNA is also a very useful tool in assessing food safety, quality, and integrity of the food chain. No, when you look at our food chain is very long. When you talk of food chain, I'm not talking about the one that the biologists will teach. I'm talking about the food chain whereby the food is being produced from the producer to the consumer. Through that process, a lot of things can be changed. And through that, we can use DNA to detect whether the food is safe. Well, when you prepare a specific food for me, I can send the food to the lab and test for the safety of the food. One, I can test the quality of the food, whether the material that we use to prepare the food is of high quality. Some of you, those of you that you sell food at the roadside, you go to the market and go and buy the rotten tomatoes and just go and nail it, everything together, and you come and use it. DNA can be used to detect, because when you use DNA, it will show us the kind of bacteria that are found in the food. And when we get that, we know that the quality and integrity of the food is at stake. We can also use DNA to also identify allergic material. You know, there are some people that when they consume specific food, their body start to react with it. So we can identify. That is why our food system here is not very strong as compared to those in the Europe. As for Europe, when you buy any food product, it is written on it that if you are someone who is lactose intolerant, when we say lactose intolerant, there are people that their body cannot break down sugar that is found in milk. So they will write on it that this particular product is lactose intolerant. So if you have that particular lactose intolerant, you will not take that particular product anymore. Then also, DNA can also be used to identify adulteration. In our food industry, adulteration is where you are using inferior goods to produce food for people to eat so that you can get a lot of profit. So instead of you to go and buy proper tomatoes, you went and buy tomatoes, the rotten one, the one that Asantis call it tomato peche. Then you use that particular tomatoes to prepare food for people. You know, that tomato is very cheap. So when you use it to prepare food and you are selling, you get a lot of profit. But DNA can help us to detect those adulteration, because we know that qualities of tomatoes or tomato that is not rotten should have this specific DNA. So we'll be able to detect all those fraud in it. And also, I have been telling people that those of you that you've been eating bush meat when you go to the market and go to buy bush meat, most of the bush meat that they bring it from the north are not bush meat. Some of them are horses. People are chopping horses here and call it bush meat. So that is what we call commercial fraud. Like you tell me that I'm going to eat grass cutter. This is grass cutter you are what is it? You are selling. But meanwhile, it is not grass cutter. It is something different. So because grass cutter is having a specific DNA, and probably a horse is having a specific DNA, when you represent one particular species with other, we will be able to detect them. So that is one thing that you need to also know. And also, DNA can also be used to identify microbes that causes foodborne diseases. And what are these microbes? We can talk of the Salmonella, typhi. All these ones are foodborne diseases. So we know that Salmonella is having a specific DNA. So when we get a food, then we test it, then we find out that this food contains Salmonella typhi. We know that this particular food can cause diseases to people, so we will not allow them to eat. So it is through this DNA that we tell you that don't eat cold food in the morning. Because when you eat cold food that contains Salmonella typhi, you will fall sick. So when you heat it a bit, we know that Salmonella typhi don't like heat. So when you subject it to heat, that particular microbe will die. And also, we can use DNA to also identify undesirable characteristics in food commodities, such as the texture. So when you cook rice and you leave it for about 3 days, you see that when rice is about to spoil, it becomes slimy. It becomes slimy. So when you see the slimy nature, you know that the food has gone bad. And the slimy nature is as a result of these microorganisms that are reacting within the food. And they will change the nature of the DNA of the food will change. And when it does that, it causes the food to go bad. So these are the few things that we can say when it comes to issue of importance of food, importance of DNA in food processing. So, any question? Any question before I move on?
Please, sir. Please, sir. I need a clarification on this point, the application of DNA in our food industry. You said that HG are double bond and they are weaker and can break down. How? It can break down by heat, meaning that they are stronger and they require high heat or what? I don't get the point right.
Which one are you referring to? You know, we have the A and T, then we have C and T.
A and T.
No, the A and T are rather the weaker. You know, when the bond,
you said they are double bound and they are weaker.
No, you see the problem. We will talk about fat and oil. When we are talking about fat and oil, once a particular product is having a double bond, doesn't automatically say that particular bond is strong. No, that bond cannot be strong. Let me put it this way. I hope you were a lady, right?
Yes.
Yes. Let's say a lady and you are dating two people at the same time, and I have another lady who is dating one person at the same time.
Okay.
Don't you think when you are dating two people, the kind of affection you are sharing among two people. So those affection between two people will be weaker as compared to the one who is dating one person and the affection is very strong.
Yes.
So the idea is not like because it is double bond, it is stronger than the single bond. No, it's not like that. In chemistry, double bond we even have triple bond. Anytime you have a product that is having a single bond, that particular product is very strong because it is stable. When you are double bond, you are stable. When you are dating two people and you want to get married, to get to a point that you are not stable, which one should I pick? But when you are with one person, you are certain that I don't have any other person at all. So probably you can give in all. So the single bond will be stronger than the double bond. I hope you are with me.
Yes, sir.
So it's good to, as a woman, to date only one or to marry only one so that you can have a stronger bond between the two of you. Nobody should talk about we guys because we will not listen. Okay, let's continue. Any other question? Any other? Topic I have about another 2 things that we will talk about. When I am done with this, I'll give you some 10 minutes break. Then we will come back and continue from the protein synthesis itself. So these are some of the things. So we can even use. Recently, some Ghanaians smoked fish was transported from Ghana to Europe, and they were rejected because the food quality was very low. Here after we smoke the fish, the processes that we pass the fish through, we don't check the quality. We just consume them. But when it comes to the high standard countries, they pass through you have to meet their standard before you can sell those products. So DNA is very, very important because when they pick some portion of the food and they subject it to lab investigation, they realize that most of them contain unwanted material and they rejected them. So it is very, very important that we trace the food from its production from the farm to the fork. When we say from the farm to the fork is when you are eating. From the farm is where it is being produced to the fork where you are eating. So a lot of things or a lot of changes can happen to that particular product, and through DNA we will be able to do it. So it's not DNA is not only to determine whether the child is yours or not. It can be used in a lot of fields. So those of you that you are thinking we'll be doing DNA tests for us to know whether the child belongs to this man or does not belong. DNA has a lot of functions, a lot. Probably when you go to the medicine and the other field too, they also use DNA in their processes. Any question? I'm done with this. We have two main units that we will talk about. I think the second unit is about 12 slides, so that way we can finish. Any question before I give you some break? Any question? Any question? Hey, Obi and Gaza. There's a Matthias here. So we are setting our questions.
Sir, please, can you go over the last point for me? Modifying the undesirable characteristic of food commodity. Can you go over that one for me?
So we are saying that we can use DNA to modify undesirable characteristics in food commodities. You know, when you pick food substances itself, when you leave certain food substances for a long period of time, a certain changes can occur, and some of the changes are desirable and some are undesirable. So through DNA modification, we can change the undesirable, or we can stop the action of the undesirable characteristics. So we've been able to learn that when you put food substances inside the fridge, the liquid in it will turn to ice. And we know that microorganisms that causes these undesirable characteristics in food cannot perform its function when we reduce the temperature of the fridge up to 0 degrees Celsius. Now, immediately a product gets to 0 degrees Celsius in the fridge, it starts to turn into ice. So those that will cause undesirable characteristics cannot work. So it means that we've been able to change it, or we've been able to stop the undesirable characteristics that may occur in that particular food product. And those who do the medicine, those who are doing the herbal medicine, when you take some of the medicine and they've been able to remove the bitterness from it, all these ones can be done through the application of DNA in our labs. Any other?
Sir, thank you.
You're welcome. Any other?
Please, sir, more light on DNA is used to identify the adaptation.
More light. Me, I don't have light. Oh, there's light out in my area. Okay.
Please.
When we talk of adulteration, the main idea should come in mind that when you are using an inferior goods to produce food to sell so that you get maximum profit. So the example that I use, I am a watch seller, or I sell rice and stew. I go to the market. Instead of me to buy the tomato, the correct one is expensive. So I went and bought the rotten one, the one that Ashantis call it, tomato peche. When they buy it, because they know what they are coming to use, they will buy it and just go to the grinding mill and put everything together and they will grind. So all the bacteria in it will form part of the tomatoes, and they will bring it at home. And they will come and cook and use it to produce food products. Through the application of DNA, when as a food biochemist, when I come and pick sample of the food, I send it to the lab and I go and test it, I will be able to identify that the food contains some certain bacteria DNA that they are not supposed to be there. So I'll be able to identify that this food contains inferior goods, inferior food substances that have been used to produce this food, which is not good for our health. So that is how it is being done. Anytime you hear the word adulteration, it means that you are using a bad product or a bad ingredient to produce food to sell so that you get a lot of profit. Are you with me? Martha, are you the one who has that?
Please, yes, sir. Understand. Thank you very much.
Understand. Okay. Any other? So I've spoken about the flood. So you are going to buy cow meat. If you are you are buying goat meat, then we give you dog meat. It is fraud. It's a commercial fraud because I didn't ask you that I'm buying dog. I decided to come and buy meat. So that is why people are very skeptical when it comes to issue of where they buy some of their meat products. When I was posted to Upper East to go and work. So when I was at Navrongo, you know, that environment, the issue of pork and dog meat is very common. I'm not saying it is a bad to eat, but me, because of my religion, that prevents me from eating those food. Anytime I'm buying food outside, I try to buy fish instead of meat because I am not sure whether the meat that I'm going to consume is a cow meat. So in order to prevent myself, because people can easily represent certain meats, and when you are eating, you might not be able to identify, and that is what we call commercial fraud. You represent a specific species with others and thinking that nobody will know. Through DNA, we can test it. We know the DNA of a dog and we know DNA of a goat. So when we put them in the lab and we subject them to analysis, you'll be able to identify that this one is this and this one is that. Any other questions? Okay, then let me give you about. It is now. What is my time? My time is 90. Yes.
The second point, identifying analytic material.
Anita, I can't hear you well.
I'm saying, please explain further.
Identify the allergic materials.
Thank you.
When you say you are allergic to something, what does it mean? When you say you are allergic to something, what does it mean? You know that we have people that when they eat peanuts. Okay. You know that we have some people when they eat peanuts, it causes some kind of discomfort to them. Some people eat. Helen, can you put on your microphone? Can you put off your microphone? Helen Ladi. Helen Ladi, you are giving me feedback. Because she's giving us some feedback. Okay, I've been able to meet her myself. So what we are saying is that, Sarah, let me finish with her then probably. Or you want to say something? Sarah, do you want to say something?
Yes, sir. You asked what this thing is. So I wanted to say that when we react after eating something,
okay,
the allergy.
I'm listening. Tell me.
I wanted to say that you asked what an allergy is. So I wanted to say when we react after eating something.
Yes. So when you react, when we consume certain food processing, food, our system react with it differently. There are people that me, for instance, when I eat cocoyam. Anytime I eat cocoyam, I will be having some burning sensation. That is how my system reacts with it. Okay, so let me use me for example. So anytime I'm going to buy a product and they've written on it, they've written on the cocoyam, it should give me an indication that when I take it, I'm going to react with it. But in the natural settings, and there are certain food products that causes a lot of reaction. People react with peanuts. I hope you know peanut, granite. Some people even react with soya. So when I go and buy food products and I bring it to my lab, and I know that when I eat peanut, I'll react with it. I can subject it to DNA test to test whether there's some traces of peanuts that is found inside that particular food product. But when you go to outside world, most of the food that causes people react with it. When you use it to prepare product, they will force you, or their regulation will entice you to write on it And the common one that people react with it is lactose. Especially people who don't consume, people that after their parents have stopped breastfeeding them, they don't take in milk product again for a long period of time. And know these things are being digested or broken down by specific enzymes. So when you don't use that particular enzyme for a long time, that enzyme will break down. It doesn't express itself again, as we were talking about gene expression. So when you start taking in that particular food, to get to a time, your body will start reacting with it. But I know someone who told me that when he or she, when she eat granite, she will be feeling some kind of pimples on her face. All these ones are allergies or reaction that our body does when you consume certain type of food. So we can use DNA to detect. So when you go and buy that particular food, then DNA can tell you that this particular food contains some allergic material. Then you'll be able to know that when I consume it, it will cause this problem to me or not. So that is what I can say. Anita, are you okay? Yes.
Yes, sir. Yes, sir. Thank you.
Yes, talk to me.
Sir, please, talking about the allergy, I think it's not only when that product is consumed. Sometimes when you just get into contact with it,
All right.
you can
Okay.
get the allergy.
Allergies. Sometimes it
Yeah.
can even be your cream. Is that also? Some people when they apply certain cream, it reacts to their system.
Yes, sir. And
So when
sometimes even the vegetables and the food products that we use.
okay,
Some people are allergic to certain vegetables. So even when they are working with it, without taking it, when they get into contact with it, when they use it, they touch it, they get the allergy. Sometimes you see some bumps on their body,
all right,
a form of rash or something. Thank you.
okay, that is good. And one thing, the reason why we are learning this is most of our food products in the market they will write it allergic free. So when you are sending this food product to outside world to go and sell, and you write there allergic free or allergen free, they will send it to the lab and go and test. When they test it and they find out that it contains some food materials that can causes allergen to people, they will just uh, they will not patronize your product. So that is what I can say about that. Any other question? Hey, people are running away, but we have a lot to do. Okay, let me pause. Let's come back 9:50. I will give you some 8 minutes or 7 minutes to rest. Maybe I'll come so that I can also take in something. Then 9:50 we will come back and continue our class from let me see what I need to. We are going to do the protein synthesis proper. Protein synthesis proper.
I say.
My headache.
I guess I may I free home. I know quite a say.
All right, so let's pause here and have some break. We will come back again. All right.
Hello.
My people, where are you? I'm ready. Come to your people and let's start. We have a lot to do, so let's start. Can I start now? Are we all in? Can I go ahead?
Yes, sir.
All right. Oh, looking at this time,
Yes, please.
can we even finish this and go to the next one? To be difficult because you have class after this, eh?
Yes, sir.
What time do you have your class?
Please, 11:50.
Is that what?
Sir, please 12.
11:50.
11:50.
12.
11:50. Okay. So let me try if
But
I
only
can use. I can use the next 1 hour.
Charlie.
I can use the next 1 hour for this particular slide. This particular unit. Probably the next unit we will combine it with what we would every week. At least we were supposed to do two, but last week we did only one. So that is the only problem. But if you are not able to finish, I'll fix a date or a time somewhere within next week or when we are supposed to, when we are about to end the lecture, then we will deal with it before. So we are using only 1 month for this. After today, we have 2 more meetings, right? So we will not
Yes,
have
sir.
it. We will handle it, no problem. So I think 25 people are here, so I think I am good to go. Okay, so this particular unit, we are going to look at proteins. We have now finished. Those of you guys are running away, master. Where we are getting to it, it's not that difficult. But I will still be using some of the structures to explain. And for the structures, you can't run away from it because you need to understand how the structures work. And because proteins perform each function based on the structure, that is why we will keep on. As for proteins, even better. When we get to carbohydrates, that one they contain a lot of structures. But don't run away. The Lord is our guide. We will surely sail through. All right. So this particular unit, our focus is going to be on the structures of proteins. And proteins have structure 1, primary, secondary, tertiary, and the quaternary. There are 4. So we will look at how the structures look like and the specific bonds that link these structures together. So you should be describing the basic amino acid structure and distinguish between essential and non-essential amino acids with examples. Then also we differentiate among the primary, secondary, tertiary structure of protein and identify the forms of modification that take place during and after protein translation. Then we describe the structure of quaternary protein. That is the highest form of protein. That is the quaternary protein. So when you say amino acids, I think by now each one of us know what amino acids are. We are saying that they are the building block of protein. They are the building blocks of protein. Remember?
Sisi koni iki mape koni ami chapone yeto.
Hey, what I can say, you know. So guys, what I can say, you know. All right. So.
Sir, sorry.
And like I over there. All right. So we will look at the building block because remember we just finished talking about how proteins are being synthesized. We said that in order to synthesize proteins, it is different amino acids that are linked together to form proteins. So amino acids basically the building block of protein. That is the foundation of proteins. Then we say that they are usually used in every cell of the body to build proteins if needs to survive. And every protein or every part of our body has some form of amino acid that have been used to build the protein. So there are a lot of protein structures within our body. And we said that, what does this remind you of? DNA. Can you say something? What does this point remind you of DNA? It's a question that we've put it over there. When we say that these are used in every cell of the body to build the protein it needs to survive. And what does it remind you of DNA? So we'll be thinking about it. And we say that all organisms need some protein, whether they are used in the muscles as simple structure in the cell membrane. So within our cell membranes too, there are some form of protein that are used. Remember, when you look at our cells, we have cell membrane and cell wall. The cell membrane are those that they are semi-permeable. They allow some certain substances to pass through, but the cell wall is very rigid in nature. So you need to just remember all these basic things that we know in science. They also said that even though all organisms have differences, they still have one thing in common. That is the need for basic chemical building blocks. We may exist in different ways. Myself as human beings, ourselves as compared to animals, as compared to plants. We may exist in different ways, but the basis of our building block is amino acids or they are proteins. That one day we can assure you of that because of the nature of our DNA and how we can transcribe them to RNA and later on translate them into amino acids and protein. So when we talk of structure of protein or amino acids, please forgive me when you hear me using protein, because I've been mentioning protein too much. But let me focus on the structure of amino acids. Amino acids have 2 carbon atoms, one of which is part of a group called carboxyl group. So the carboxyl group is the one that we've indicated as COO minus. The minus is there for something. I will talk about it. COO minus. So in chemistry, when you lose an electron, you become when you gain an electron, you become negative, and when you lose it, you become positive. And we said that the carboxyl group is made up of 1 carbon and 2 oxygen atoms with a negative charge. So this is the structure. We said that the carboxyl group has a negative charge since it is a carbolic acid that has lost its hydrogen. So under normal circumstances, you see, I think you can see my case that COOH, when it starts or when it forms, when it come and join the amine group, protein, we have what you call the carboxyl group and the amine group. So the amine group is where my case is, and the carboxyl group is the one that is top here. So it is because of the COO minus that is making it more acidic. So this particular H, the structure, this H was part of the carboxylic group, but when it comes to react or during the reaction, the COO lose the H to the amine group. Then now here will become N and 3H. So that is how it's supposed to be. And we are saying that the second carbon is connected to the amine group or the amino group. And amino means that there's 1 nitrogen and 2 hydrogen group bonded to the carbon atom in the image below. We said that these positive and negative signs are there because there is a donation and recipient of electron in the compound. Let me not go a lot deeper in this chemistry before people run away and leave me. So what we are saying is this amine group or this acidic group, let me put it this way, the carboxylic group and that of the amine group, when they start reacting with each other, there's one H here. You see, when you see the structure COOH, when immediately come in contact with the amine group, the H here will be lost and be given to the amine group to form the amino group. So that is what we need to know. At the normal circumstances, it's supposed to be NH2, but here you can see that the H is 3, 1, 2, 3. So I'm just using the structure for you to appreciate what it means. And we said that the negative and the positive signs are there because there has been a donation and recipient of an electron. When you donate something or when you lose something, you become negative, and the one that gain become positive. So that is basically what it is. And we said that 1 hydrogen atom moves to the other end of the molecule to form carboxylic acid, which give it positive charge, while the giving away results in negative charge. I think I have made mention of this. So amino acids, the side group are what make each amino acid different from other. There are two main side groups out of 20 amino acids used to make protein. So the first one, we have the polar side and we have the non-polar side. I don't know whether when you were doing interscience, you spoke something about hydrophilic and hydrophobic nature of. Did you talk about it? Have you done anything? Have you done something like that? Okay, so don't worry, we will take our time and look at it. So the two main groups are the polar and the non-polar. When we say something is polar, something that is water loving, or that's something that want to react with water. And the non-polar is the one that does not want to react with water. They are more of fats in nature. You see, no fats and water doesn't meet. Anytime they meet, there will be a sort of repel. There's always a separation in it. So in chemistry, we term them as hydrophilic, that those that like water, and hydrophobic. Hydrophobic are those that fear water, those who don't want to associate themselves with water. And most of them are fat in nature. So that is, and we say that this polar, the polar side, we say that this refers to a way the side group, sometimes called the R group, radical group, interact with the environment. Polar amino acids adjust themselves in a certain direction. So anytime amino acids find itself in an environment where it is water, the polar side of the amino acids start to react with the water, and the non-polar side will try to coil inside because it finds itself in water. Because non-polar don't want to deal with water, they try to coil themselves. They have to hide that particular side so that they will not get any reaction with water. They will say that the non-polar side is a way that the side group don't really care what is going on around the environment. That is, the non-polar side do not adjust to the environment. The polar and the non-polar chemical trait allow amino acids to point towards water. That is hydrophilic. Anytime you hear the hydrophilic, water loving, or away from water, hydrophobic. So I can ask you to differentiate between what hydrophilic and hydrophobic is. Proteins, the part of protein that are hydrophilic will always want to react with water. And those that are hydrophobic will always want to run away from water because water and fat doesn't meet. So that is basically some of the things that you need to just understand. We say that these characteristics of amino acid chain facilitate twist and turn of growing chain when they are being synthesized. Please, a minute, I've gotten I need to add you to something. A minute. All right, I'm back. Sorry. So we are saying that the characteristics of amino acid side chain facilitate twist and turn. So when we start looking at the structures, you know that at a point in time, some of the amino acids, the protein structures will be turned, or it will be twisting, it will be folding because of this characteristics nature of it. There is that out of over 50 amino acids discovered by scientists, only 20 are used in synthesizing protein. So in the beginning, I told you that there are a lot of amino acids that have been discovered by scientists, and only 20 of them can be used to synthesize proteins in the body. Only 20. And even with the 20, there are some that the body can produce them and some that we need to consume them as part of our food. This one there, I can see it is going smoothly. That's also, is it not moving smoothly? Moving.
Yes, sir.
Is it not
Yes.
moving well?
Yes, sir.
Yes.
Sir, it
Yes,
is
sir.
raining at my end, though.
It is raining at your end.
Heavily.
Don't worry. Then you are blessed. All right. Let me continue. So when we want to classify amino acids, we classify them based on two main things. We have the essential amino acids, and those that we call them the non-essential. When we say something is not essential, it doesn't mean that they are not important. And when we say something is essential, it means they are important. When you say non-essential, in a layman's view, they are not important. But when you come to in our chemistry terms here, the essential ones are those probably that the body cannot produce them. Those that the body cannot synthesize. Let me use the specific word, synthesize them or produce them. And the non-essential ones are ones that the body can produce. They are non-essential because if you don't even consume them as part of your diet, your body can still synthesize those proteins. So we call those ones as the non-essential. So the essential one, they include histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, and valine. Then we also have what we call the adenine. Essential in children but not in adults. So what it means that when it comes to children, they are essential, but in an adult, they mean that in an adult they are non-essential. The body can produce them. Then the non-essential, we have the alanine, we have the aspartic acid, glutamic acid. Then there are some that we call them conditional amino acids. So under certain conditions, certain non-essential amino acids become essential under certain conditions. An example of those ones, those amino acids are the tyrosine, the cysteine. And when you give birth to a preterm child, a child who doesn't spend all the 9 months in the mother's stomach, or a preterm child, those preterm children, they don't have, the body cannot produce the cysteine. Cysteine is supposed to be a non-essential amino acid, but the body cannot produce them. So at that moment, that particular child, you need to add cysteine as part of their diet so that they will be able to survive. Not only preterm children, there are different instances whereby a particular non-amino acids become essential. Probably you can read on it then and understand. Then also we said that we have thousands.
Please,
yeah.
Please, where will you get cysteine?
Yeah.
For instance, if you want to give it to your child.
So we'll find out, look for a specific food that contains cysteine. So this is why you need to do something that you can just Google and you will get it.
Okay, sir.
I may not have it off head like when you eat tomatoes or you eat this, it contains cysteine, but when you type this thing online, you can read them and you get it. Yeah, somebody wanted to ask us.
Sir, please, it's me. Say about the preterm child that you are talking about. I have one. That's my boy.
Okay.
So I wanted to know more about it.
Yeah, don't worry. You want to come and see dietitian and pay for consultation. You cannot seek dietetic help in class.
Okay, sir.
We can talk about that one a back door so that you can do some small payment. Is that okay?
Okay, sir. Thank you.
I need money. I will charge you D-Levy. All right.
Yeah.
So this is the structure of all the 20 amino acids. This is the structure. So remember one of the slides I told you that the only difference that exists between the amino acids are their side chain. So the color blue that you see represent the side chain. So when you look at amino acids like glycine, the glycine or the glycine, the side chain is only hydrogen. But when you look at the next one, alanine, it is CH3. It's CH3. So CH3 glycine methyl, probably. Then when you look at the valine contains H3C bonded to carbon, another bonded to hydrogen, that is bonded to methyl group again. So you can see that each of their side chain is different, and that is what differentiates one amino acid from the other. If not, the other component is the same. The other component is the carboxylic group and the amine group that I spoke about when I was in our previous slides. So this picture is there. Don't go and memorize this thing. I don't think any one of you can even memorize this thing and keep it off it. If this is the only exam for you to pass to go to to go to and teach in the university, it will still be a problem. We are not expecting you to memorize it. The picture is there for you to appreciate that all protein structures are differentiated by the side chain. So the kind of side chain that give it the specific differentiation. So that is what I want you to appreciate here. Now let's start with our protein structures. There are protein structures. Because we know that most of our enzymes, most of our hormones, a lot of the body tissues are made up of proteins. We need to understand the structures and how they are linked together. So the first one that we were looking at is the primary structure of the protein. Primary structure. So when you have valine glutamic acid, then you have proline that are linked together. This is a basic structure, a basic structure. That is different amino acids that have been linked together. Probably the specific bond that link this one together is probably what we call the peptide bond. And you need to also take note of the bond that hold these structures together. So under the primary protein, the bond that mostly exists between them is the peptide bond. So you remember our basic science whereby we used to talk about protein will be converted from protein to peptide. Before peptide will be converted to amino acids. So all these ones are something that we had a specific knowledge on it basis before we even got here. And we are saying that the primary protein is a linear sequence of amino acids in a polypeptide chain. When you hear the word linear means that a straight chain. It doesn't have branches. It is straight. It may trace along the way, but the chain is always linear. So let's see. One of the band person was starting in school from university.
Maybe I'm going to yaro mo kan yi store. Good foul through ben yo. Man je yin nga ja on ba.
That's the thing. Hey Mansura. Then you are telling him, all right. So let's look at this. Let's look at the essential amino acids. We have histidine, we have isoleucine, we have leucine, then we have lysine, methionine. When I link these things, all this one together with a peptide bond, then I say that I've gotten a primary structure of a protein. And I'm saying that it is a straight chain sequence. So it is just a straight chain that has been linked with a peptide bond. So that is what you need to understand. And you can see that the example here, we say that it is repetitive spatial of two-dimensional arrangement. That is a 2D arrangement held together by covalent bonds such as peptide bonds, which are made during the process of protein biosynthesis. So the peptide bonds are able to link them together when you are synthesizing protein. That biosynthesis, biological synthesis of protein. That is what it means. We say that the two ends of the polypeptide chain are referred to as the terminus, and the name based on the nature of the functional group in each extremity. So you can see that we have the terminal, the N terminal, and the C terminal. So that is where if another one want to continue, it will start bonding. It will continue to bond till we get a specific chain of amino acids that we are looking for, or the protein that we are looking for. We are saying that the carboxylic terminal, that is CH, is the residue in the peptide that has a carboxylic group. So always the terminal, the C terminal, you can see that this is the C terminal. That is where the carboxylic group is COO minus. Remember, I told you that the carboxylic it was supposed to be COOH, but because it has lost one hydrogen to that of the amine group is now COH minus. So have it in mind. And the amino terminal, that is amino site, is a residue in the peptide that has amino group. So you can see NH3 because it has gained one hydrogen electron. So these are all still describing how the structure is. So here, the key thing that you need to pick from here is primary protein is just a long chain of different amino acids that has been linked together by a peptide bond. So basically, that is what you can pick from this particular point. So remember the peptide bond and the linear sequence of amino acids. So we say that by convention, the primary structure of protein is reported starting from the amino terminal, which is the end where the amino group is not involved in the peptide bond, to the carboxylic terminal. So this is where the amino terminal is. Let me go back. So you can see the amino terminal. This is where it is. It is bonded to the CH here. So most of the time it started from here and it moved, and always the carboxylic terminal become the end. So if you get another amino acid that want to join, it will come and join from here, then the carboxylic group will move to the end part. So that is how the reaction always occurs. We are saying that in the biological translation, amino acids are polymerized via the peptide bond to form a long backbone with a different amino acid chain protruding along it. So when you hear the word polymerize means that you have a different amino acids. When they come together, it come and form polymer. We have monomer and polymer. So monomer is just the single one. When a lot of monomer come together, they come and form what is polymer. So what we are saying is amino acids are formed via the peptide bond. So it is when I have one amino acid and I'll join another one by peptide bond, then another one will come and join using the same peptide bond. So it continues until I get a long chain of amino acids. And we say that the primary structure of a protein is determined by the gene corresponding to the protein, and this can be directly sequenced of or inferred from the DNA sequence. I think based on the DNA that we've done, you should be able to know. So what they are saying that the primary structure is determined by the kind of gene or the sequence of DNA that encode it. So if you have a where did I write? I wrote that somewhere. If I have a gene, it is proline. So this gene will give me the primary structure. So it will determine by the kind of gene that has been expressed. And we said that we can get this one from when we are doing the DNA. So when it's removed gene CC, it will code for different amino acids. CCU, it will code for different amino acids. So that is what we just did before we came here. We said that the sequence of protein is unique to that protein and define the structure and the function of the protein. I think I have consistently said this, that the function of a protein is determined by the structure. When you change the structure of a protein, it cannot perform its functions anymore. And sometimes we alter the function, we alter the shape of a protein to perform different function aside its primary function that we want it to perform. All right. So we said that peptides can also be synthesized in the laboratory. So the peptides, those peptides that form within the protein, we can synthesize some in the lab through the chemical processes. We can also generate some of them. We say that chemically peptides are synthesized by a range of laboratory methods, but in opposite order to the biological synthesis. You know, the biological synthesis starts from the N terminal, and it starts with the N and end with the C terminal. But when you want to synthesize peptides inside your lab, you rather start from C terminal and end with the N terminal. That is why we said that they do it in an opposite order to that of the biological way that we do it. So this is the primary structure that we can talk of. So you see that glycine, if I can see the glycine, proline, whatever. These are all different amino acids that have been linked together by a peptide bond. a peptide bond. So different amino acids have been linked together by a peptide bond. So the structure is just for you to appreciate. And we said that it is totally recommended to use the word amino acid residue when discussing protein because a peptide bond is formed, a water molecule is lost, and therefore proteins are made up of amino residue. Like how should I do chemical reaction online? My people, how do I do chemical reaction online? How do we do this our reaction online? So I have my pen and paper here. Yeah, Sarah.
Maybe you do the demonstration one day and then we'll observe.
You observe from where?
From on Zoom. Then we'll all.
So I have to on my picture or what? My
Yes,
camera.
sir. Yes, sir.
I'm not wearing clothes. So
then maybe when we meet next week, you can quickly do that.
No, let me try and do it because we are meeting online throughout our lecture. So let me find a way to do this reaction. You understand?
Okay, sir.
Let me come back and look at it.
Yeah.
Remember the reaction of we said that the point is trying to let me explain the point first. The point is saying that it is recommended to use the word amino acid residue when you are discussing protein because when a peptide bond is formed, a water molecule is lost, and therefore proteins are made up of amino acid residue. Does someone understand this point? Here.
No, sir.
Deborah, it doesn't mean that nobody understands.
Please, I think that the water molecule is lost. The residue is basically, let me say, a solid.
Okay.
So it means it doesn't contain any moisture. The amino acid doesn't contain any moisture. That is when the water molecule is lost. That is my understanding.
Okay. Any other understanding? Any other point? All right. So let me quickly talk about it. When we say amino acids, when you are talking about proteins, we don't say amino acids, but rather we say amino acids residue. You know, when you look at the structure of this amino acids, I told you that it has carboxylic group, which is made up of CO. Who remember CO what? CO. Let me see that.
Sir, please, this CO2.
No.
CONH.
CONH, good. CONH. That is the carboxylic group. Then it is going to react with the amine group, which is NH2, right? And you know, chemical reactions, these things, we call them the reactant. The two compounds here are reactant. Then they are going to produce what you call product. So in the product, this one is going to produce what you call it's going to produce water, which is water is H2O, right? It's going to produce water. Water is going to be lost. And once water is lost, it means that what has formed the protein is not the actual amino acid, but a part of amino acid. That is why we call it residues. Some portion of amino acids is one that has formed the structure of the protein, but not the entire amino acid itself. Am I making sense? Am I making sense?
Yes, sir.
So what it means is, let's say we have the carboxylic terminal of the amino acid COOH. That is the carboxylic terminal plus the amine group, which is NH2. And when these two things undergo chemical reaction, we have COOH. I'm writing them, although you to write them. We are doing chemical reactions or balancing of equation online. It's not my fault. So we have to force and understand. So COOH, that is going to react with NH2, the amine group. So we are going to get we are going to get CONH plus water, which is H2O. And we say that anytime you are forming peptide bond, this water is lost. We give this water away. So when this water is lost, it means the protein structure is not made up of amino acids, but rather amino acids residue, some portion of amino acid, because the water is no more part of the amino acids again. Is it clear? That is what the point is trying to tell you. Okay, it is not a main point, that's something that we will, but just want you to understand that within the chemical reaction, anytime peptide bond is being formed, water molecule is lost. Anytime peptide bond is formed, water molecules is lost. So that is, and once water molecule is lost, it is not the amino acid itself, but some residue of amino acid. That is what is going to form the protein. So you just have to understand that. I've done my chemical reaction in my book in front of me, so I'm expecting you to do yours in front of you. Amen. All right.
No amen, no, sir.
You understand? Don't worry with this. At the point that I want you to pay a lot of attention to the structure, I will let you do that. But this one, I'm just using it to just explain for you to appreciate that proteins are made up of amino acid residues, but not the amino acid itself. So just have that particular thing in mind. Okay. So now we said that with the protein structure modification, sometimes we modify protein structure to perform certain function for us. Sometimes that is what we call mutation can occur within the protein system. So once I change the protein structure, when you modify it, it come and perform specific function. So when you have the hemoglobin that have been used, immediately you change the structure, it cannot perform the functions again. It cannot perform the functions again. So the sickle cell that I was using last week, the sickle cell structure is supposed to be biconcave or round in nature. When we say a child is having or suffering from sickle cell or someone is a sickler, there's the basic problem the person is having is instead of glutamic acid, I think I have that particular thing. Let me see so that I will show you. I think this is. Have you all seen the structure on your screen? Have you all seen it? Have you seen it?
Yes, sir.
So this is a normal DNA sequence for hemoglobin A. Hemoglobin A. That is a normal hemoglobin. So you can see that we have thymine, we have guanine, we have alanine, and its corresponding bases, alanine, cytosine, and uracil instead of thymine, because this one is in RNA, uracil. There's a second one. So the protein, the amino acids that are involved is proline, glutamic acid, and another glutamic acid. But when you come here, there has been some change. That is what we call mutated DNA. There has been some change in some of them. So instead of glutamic acid here, it has been replaced by valine. So immediately it was replaced by valine. The size or the structure of the hemoglobin changes from its normal biconcave shape into a circle shape. A circle shape is like when we sight the moon first, you look at how the moon is a crescent. And when it becomes like a crescent, the size has become very small. So it cannot carry oxygen as usual. At first, if the round shape can carry a lot of oxygen, now the size has reduced, so we cannot carry oxygen. That is why you see those people, they are always, you see them like they are always sick. So you can see that because only one part of the primary structure was changed, it has affected how the shape look like, and also how it can perform its function has also changed. Please, you understand the concept I'm trying to paint here?
Yes, sir.
All right, then that is good. I just want you to use that to appreciate what I'm trying to teach this morning. So we say that protein structure modification, there are two forms of protein structure modification, namely translation and post-translation modification. And these are considered as part of primary protein structure. So let's look at the translation modification. So we say that this form of structural modification occurred during protein translation. And who remember protein translation? Who remember? We just did translation and transcription about 30 minutes ago. Who remember that? Protein translation. Who remember that? The time is almost up. I have to be very quick, but I don't want to rush on the issues. Who remember protein translation? Yes. Yes, Esther. Esther, what did I say protein translation is? If Esther is not talking, then Vivian.
Sir, I remember that you said when we use the RNA information, that is the messenger RNA, the information it carries, when you use it to do the protein synthesis, it means you are translating it.
All right. I think that is a basic explanation that probably you can also give. Yeah, that is good. So we are saying that protein, what is it? The cross-linkage modification mostly occurs during the translation phase. So when we are doing the translation is when you are synthesizing protein from the information that the messenger RNA bring it from the nucleus into that of the cytoplasm. So there can be some form of modification. And we say, for example, when you have insulin, which is composed of 51 amino acids in 2 chains linked by disulfide bond. Remember, I spoke about the insulin. I said that the insulin is having, I think it is having 51. So we have 21 on 20 on the alpha chain and 31 on the beta chain. So there are 2 chains that come together to form what insulin is. So we are saying that they are linked together by disulfide bond connecting 16 molecules. One chain being 31 amino acids. So the 31 is the beta chain. Then you also have the 21 alpha chain. So if you want to do some kind of modification here, we can easily change the beta chain to 30 and the alpha chain will be 21. When it does that, it means that there has been some form of what is it? Some form of change or mutation when it comes to the chain that the protein contains. And when that one is particularly done, it is going to change the function of the protein. So that particular insulin cannot perform this function that it does. No, insulin mostly helps your body to what is it? To metabolize glucose or to pick glucose from your sugar. So that is basically one thing that we can know. So know that with the cross linkage, we are looking at when the translation is done, the modification is done during the protein translation process, during the time that the protein is being formed from the messenger RNA. Then we also have what you call the isomerization. So put this isomer, we will meet it somewhere when we are doing our carbohydrates. So put it in my isomer. So we say that a molecule that exists in 2 asymmetric form, although this does not change the sequence of amino acids, it does affect the chemical property. So when you hear something is isomerism or isomer is that when you have a specific molecule that when you divide it, you are going to get equal parts. So That is what we mean an isomer or isomerism. And we say that this does not change the sequence of amino acid sequences. It does not change the arrangement of the amino acids, but rather it will change the chemical properties. And what are the chemical properties of this protein? The chemical properties are the code that it contains. And when one of the code changes, so let's say if you have gene, which will code for what is it, proline, and you come and make it GCG. You have not changed the sequence, but the chemical property inside it has changed. And once the chemical property changes, you are also changing the the function of that particular protein. Then the different way that you can also make modifications is post-translation. Post means after the translation, after the formation of the protein. We say that this form of modification occurs after protein translation. Example is phosphorylation. When we talk of phosphorylation, in the process of producing energy in the body, there are glycolysis, phosphorylation, pentose pathway, and the rest. There are a lot of processes that the body uses. So one of the ways that we can do modification is phosphorylation and also glycosylation. That is when you are breaking glycogen to yield the kind of energy we are looking for. We say that protein phosphorylation is one of the most common occurring and most studied post-translation modification. It is the addition of phosphate group. So when we say under the phosphorylation, you are adding some phosphate group to the protein, phosphate group to the protein structure. An example, we say that it is normally occur at the serine, tyrosine, proline residues. So these are some of the specific amino acids that post-translation modification can occur. And specifically phosphorylation, when you are adding phosphate group to that of the amino acid. It can also occur at the same time when you are adding some glycogen or glucose molecules to that of protein. That is what we call the glycosylation. We say that it makes the amino acids transition from being hydrophobic to hydrophilic, enable it to interact with the environment. So here, this is the key thing here. Probably you may find a specific protein that is all these protein that we made mention, the serine, the proline, the tyrosine. If you look at their side chain, most of them they don't like water. When they find themselves in water environment, they don't want it. They become unstable. So if you want to stabilize them, then you can add the phosphate group to it. When you add the phosphate group to these particular proteins, they become more stable, and they begin to love the water environment that they find themselves. So these are some of the idea that we should use. We do to make protein stable in certain environments when when naturally they are not stable in that particular environment. Then we can also I think that is basically about the primary structure. So here, know what post-translation means and at what point do post-translation occur. The type of post-translation that occur are two: phosphorylation and that of glycosylation. The phosphorylation is you are adding some phosphate group to proteins to make it more stable in specific environment, and glycosylation too is the same. You are adding glycogen or glucose molecules to a specific amino acid to also make it more stable in the environment that it finds itself. Now let's look at the secondary structure. We are finished with the primary structure. We've looked at the various components of the primary structure. I think our time is almost up. Let me quickly before I end this class so that you can prepare for the next. So that the secondary structure of the protein refers to the highly regular local folded substructure that are formed within the polypeptide backbone. The secondary structure of a protein is due to the folding polypeptide chain into different folds due to the hydrogen bond and van der Waals forces. So now pay attention. With the primary structure, it was just individual amino acid that has been linked together by peptide bonds. But when you come to the secondary structure, it is the primary structure. You may have 2 or 3 primary structures that are linked together by hydrogen bonds. Hydrogen bonds, or what we call, or another forces that is what we call the Van der Waals forces. But our focus should be here that you have about 3 or 4 primary structure that has been put together and linked by the hydrogen bond or the Van der Waals forces. And we say that the secondary structure is mostly folded. They're linked to one another. I think I have a picture here. Let me bring it up for easy explanation. So you can see that this blue is one primary structure, the second one is another primary, and the third one is another. And they have been, this small dot that you see here are the hydrogen bonds or the Van der Waals forces that I'm talking about. So you can also see it here. So you can see one primary structure, another primary structure, another primary structure, and they are being linked together. So we have the alpha, the alpha helices and the beta pleated sheet. So let me still use it too. So you can see that this is the beta pleated sheet. They have done it in the form of a sheet, and this one is the alpha helices. So you can see that this one is just coiled. It is just coiling to each other. That is the alpha helices, and we have the beta pleated sheet. So the beta pleated sheet is more like you have 3 different strands that have been linked together by the hydrogen bonds or the Van der Waals forces. And we say that the secondary structure are identify the patterns of hydrogen and disulfide bonds. So you see the bond that are forming the secondary structure, we've made mention of hydrogen bonds or the Van der Waals forces. Then we also have what we call the disulfide bond. Disulfide bond, that is 2 sulfur molecules that has been linked together. It becomes or the sulfide bond. So that is another bond that also occurs when we are looking at the secondary protein structure. And we say that the most common secondary protein structure are the alpha helix. So this one is the common one. This one, the one in red. So it's the same thing that they've replicated here. They are more common than any other secondary protein structure that you can talk about. And we say that both alpha and the beta sheet represent a way of saturating all the hydrogen bonds, donors, and acceptors in the polypeptide chain. So basically, this is something that we need to also know. Then we say that the structure, the sequence of the variable group and how they interact with each other through hydrogen and sulfur bond determine the secondary structure, whether it is going to be alpha helices or they are going to be beta pleated sheet. So the sequence, the way they are going to do it, when it is spread in 3-dimensional way or 3 strands, it is going to be the beta pleated sheet. But when it coil like this, and the way these shapes are being done it is not just done by any way. It is the interaction between the hydrogen and the disulfide bond that are forming. So the way the interaction is going to be determine how the sheet is going, the specific shape is going to be. And this interaction is being done by the donation, how one donates and other one also accepts. It involves donation of electron and acceptance of electron throughout. And we said that this tend to help to determine the tertiary structure and overall shape of individual protein and its functions. So the insulin that I was talking about, this is how it looks like. This is how it looks like. So you see, the bond here is sulfur-sulfide bond that is holding them together. We have sulfur-sulfide bond, that sulfur-sulfide bond, and the hydrogen bonds are holding the individual primary structure together. So this is the typical structure of insulin, the one that helps your body to absorb glucose, the glucose molecule into your tissues. So this is how the chain look like. We have 21 chain, we have 30 chain, alpha and beta, if you remember. Then from there we talk about the tertiary structure. So the tertiary is more of 3-dimensional native structure of a single polypeptide or protein. A protein normally folds into compact structure, usually referred to as the globular protein, a term traditionally associated with water-soluble proteins. The secondary structure are stabilized by the final native fold. So with the tertiary structure, when you pick the secondary structure, the tertiary structure tries to fold to become more globular. Globular is becoming more round. It means that the folding continues to be many in the tertiary structure as compared to the secondary structure. The tertiary structure, the protein is going to fold more. I saw one structure here. Let me see if I can use it to make it. So you see the tertiary structure. The primary structure here is just a linear of amino acids, different amino acids that has been joined together. Then when you come to the secondary structure, we have the alpha helix and the beta pleated sheet, the one that I spoke about. So when the bonding becomes so many, it starts to form a rounded figure. It starts to form a rounded figure, and that is what we call the tertiary structure. It becomes more globular, become more rounded form. Then when that different tertiary proteins also come together, it will come and form what we call the quaternary protein. So the quaternary protein becomes the highest structure of every protein system that you can talk of. So let me come back again. We said that the alpha helices and beta pleated sheet are folded into a compact globular structure driven by non-specific hydrophobic interaction. So there is the interaction here is hydrophobic. It is not hydrophilic, it's a hydrophobic interaction. Hydrophobic means that those interaction does not involve water. When it is hydrophilic, it means that the interaction involves water. By hydrophobic, it means that those interaction does not involve addition of water. And we say that the tertiary structure has a simple polypeptide chain backbone with one or more protein secondary structure. So we can have, let me come back to the structure again. So we can have one particular simple primary structure with so many secondary structure that has been linked to the protein, the primary structure to form that particular the tertiary structure. And we say that amino acid chain may interact and bond in several ways, and this interaction and the bond within the particular protein determine its tertiary structure. So you can see that there may be one. Let me use it. There may be. Let's use this instrument. There can be this particular red here. This particular red is another amino acid. It can just come here and come and bond with another red substance here, probably one of either valine, glutamic acid, or whatever. So the bonding becomes too much within itself to the point that it will start to form a globe-like nature. So that is what we can pick from the tertiary, and then the quaternary protein. The quaternary, that is the last form. So most of our protein structure in our system are made up of quaternary protein. Most of our protein structure, the hemoglobin I was making mention of, the ribosome, the antibodies in our bodies that fight against, in our immune system that fight against disease are all forms of quaternary protein. So we say that it is when individual polypeptide chains associated with each other to form a larger protein complex of geometric specification arrangement. This only arises when the protein is made up of two or more polypeptide chains. So you see that when you have two or more, polypeptide chain is the backbone of protein formation. So when you have two or more polypeptide chain, remember I told you that with the tertiary protein, it's made up of one polypeptide chain with different either alpha helices or beta pleated chain joined together. But when you come to the quaternary, it is made up of more than one polypeptide chain, and this is the polypeptide chain that I'm talking of. This is the polypeptide chain, the primary structure, the backbone. That is the polypeptide chain. So when you have two or more polypeptide chains that have been folded together and is driven or reinforced by the formation of many bonds between different parts, the individual polypeptide protein complexes are referred to as subunits. So the individual polypeptide chains that come together to form the quaternary protein, we call it the subunit of the polypeptide chain, because if you have 3 or 4 polypeptide chain, one of them will become a subunit of the polypeptide chain. An example is the hemoglobin, your DNA polymerase. Do you remember DNA polymerase? When you are forming,
Yes, sir.
when you are adding the corresponding base to the DNA during cell divisions. So that particular one too is made up of protein. They also have ribosome. The ribosome itself, I told you the ribosome is where the protein synthesis occurs. They also have the antibodies, a portion of our white blood cells that fight against diseases. So basically, this is what we need to know under this. And we said that the rules of function of the structure, protein structure. So we said that the function of the protein determined by its shape. So when you change the shape of a protein, it cannot perform the function again. I think I have made mention of this over and over again, and it's so important. I would say that the shape of a protein determined by its primary structure, that is the sequence of amino acids. If the primary structure changes, the shape of the protein can also change. And we say that the sequence of the amino acids in the protein is determined by the sequence of nucleotides in the gene. And what are the nucleotides in the gene? Either the adenine, the cytosine, the guanine, or the uracil. The sequence of the amino acids are also be determined by this kind of nucleotide. So this is what we need to know when it comes to the structure of protein. And basically, this is the structure. Primary, which is made up of only the backbone, polypeptide backbone. Secondary, which is there is more interaction between the individual amino acids. Then we have the tertiary. Tertiary one contains one backbone and two or more secondary protein that are interacting with each other using hydrogen bonds and the hydrogen and that of the sulfide bond. Then we can have the quaternaries, which is having two or more backbones, which is trying to interact with each other to form a more complex protein structure. So these are how the structure of a protein look like. I think I've used this example already, so there's no need. And our time is 11 o'clock. I don't think we can do the next one that I want us to do. So I'll end the class here. I'll invite questions. Then the next thing that I wanted us to look at is the application of this protein structure in food processing. So I'll send you the slide so that you read ahead. It's only 12 units. So the next time we will meet, we can quickly start from this one. We will finish with that one. Then I will start with the carbohydrates. And that one too is a lot. So if you have any question, I think probably you can bring it for us to look at them. Any question?
Sir, question.
Yes.
Sir, please, can you kindly go back to the structures, the primary, the secondary, the tertiary, and the
Then you are telling me to talk about all of them again.
No. I want you to please the slide so that I will ask the question on it. Please, sir, are you saying that when it comes to the primary structure, it is made up of individual amino acids coming together to form the structure of the primary structure?
Very good.
Then, when you come to the secondary structure, it is made up of only individual amino acids, only structure, which then becomes the alpha.
Yes, you have a point. The primary structure is just basically.
Sir, the slide is gone.
The slide is gone. Yeah, I can see mine.
Sir, it's back. It's back. And then, sir, when it comes to the tertiary, are you saying that one you have this primary structure, that is the different amino acids coming together to form one backbone, and then the secondary structures, which is the alpha and then beta sheets coming together? So the tertiary is made up of one backbone, then the alpha, and then the alpha helix, and then the beta pleated sheets.
Yeah, the interaction between them will come and form plus the primary structure, plus the polypeptide. You know, the polypeptide, the bonds only exist between one amino acid and the next amino acid. So if it is
polypeptide,
so that is the polypeptide bond. It's formed between them. That is why it is arranged in a chain-like form. But when you come to the secondary, the bond moves beyond only one amino acid and the other. there may be one amino acid that last year
Hello,
can
sir.
react with the first one or the middle one.
Hello.
Hello. Can you hear me?
Sir, you said that when it comes to the secondary, the bond will do what? The primary bond.
The interaction is not only between one amino acid and the subsequent one or the one that
Madam
follows.
Sarah, please. Madam Sarah, please mute your mic. Sir, the secondary, you said it's not made up of only one amino acid.
No, I'm not saying that it's not made up of. I'm talking about the interaction, the bond that is formed. You know, when you get to the secondary, the bond that is formed is not only the peptide bond, but also there's also sulfide bond. I hope you get it. So it is through that particular interaction that makes the structure look like this. So let me bring this. So have you seen this picture? So these ones are all individual amino acids, but look at how the interaction is being done. There are a lot of bonds that is forming in between them. And the way the bonds are formed determine the kind of shape it is going to take. Because there are instances whereby a bond can be formed from the first amino acids into the last amino acids. And when it does that, automatically it is going to coil the structure. But when the bond is formed in the form of a mesh, so the beta pleated sheet, the bond, the interaction here is in more of a mesh form. This one join this one, then this one will come and join this. So you can see that if this one is one amino acid, it is linked to another amino acid, and the same amino acid here too is linked to another amino acid here. So you can see that the bonds formed between almost all the amino acids, they interact with each other. But when you come to the primary structure, the amino acids, the first amino acids reacted only the second amino acid. The second one reacted only the third. The bond formed between them like that. But when you get to the tertiary stage, let me go to the tertiary stage, it uses one backbone. Then different alpha helices or beta pleated sheet. Then another interactions will be moving within them so that it will become more of a globe. When you say globe, in a round form.
In a round form, yes, sir.
Then from there, if you want to form a quaternary structure, the quaternary structure uses about 2 or more of the
tertiary
polypeptide base, the backbone.
Yes, sir.
So when 2 or more polypeptide backbone come together, you can see that we have a lot of different amino acids.
Yes,
well, if one polypeptide chain, let's say one polypeptide chain contains about 20 amino acids, then you have 2 or 3 coming together. It means that it is going to form a lot of bonds. And the more, when I bond with you, I get closer to you. I pull you along me. So each of them will be pulling each other. That is what makes it form this particular structure that you see.
Okay, sir.
Is that okay?
Yes, sir. So, sir, it means that the secondary, it is bonded by polypeptides and then the sulfide bonds.
I think when you read your notes, those things are explicitly stated over there.
Sir, I'm coming on.
All right.
Sir, that one, the structure, the structures. So when you look at the structure, say the secondary, it means that when you get to the tertiary structure, there is no polypeptide bonds or the sulfide bonds there. That is what I want to ask.
I don't. There's no peptide bond like how?
The peptides and the polypeptides and then the sulfide bonds that is in the secondary structure. Does it also follow to do that in the tertiary structure?
Yes, it is there. It is there.
Okay.
It is there because if there is a lot of polypeptide bond in the secondary, as for the bonds, they are there. Just that at the primary level, it is only a peptide bond, but from primary level up to secondary, tertiary, and quaternary, there's an addition of disulfide bond and also, we spoke about the Van der Waals forces. All these ones are forces that pull these proteins together. So those bonds are still there, but they become more because the more you have protein structure or the more individual amino acids involved, the more the bonds are going to be many. So when we were in primary school, do you remember there was some time that we will form a circle and we will pulling each other towards ourselves. Do you remember that? It is like those forces pull us together. So the more you have a lot of people pulling together, the more we are able to form a proper bond within ourselves. So that is what the idea that you should just picture, and I think we will be fine to go. Hey, people are running away. Any other question?
Yes, sir.
Any other before I leave you to go and do your. I think you have microbiology, right?
Yes, sir.
Yes, sir.
Don't worry, it will be fine. It will be fine. Israel will handle you well. So next week we will continue. We will do the application of these protein structures. Then we will continue from possibly our carbohydrate. There are a lot of structures that we'll be looking at. So just appraise yourself and you understand. Read the slides. If you have any challenge, my number will be on the slide, or you can contact us. And I'm even on the platform. When you check the names, there will be a name called Kimbawa. That is me. You can WhatsApp me or call me and I'll respond to you. Yes, Ramatu. Yes, Ramatu, your hand is up. Can we hear you?
Sir, please, I'm not.
You are what?
Yes, sir. Sir, please, I'm not on your page, and I still need your number.
It is
The
not
whole
my
thing is
page.
confusing me. Oh, please, I'm begging you.
It is not my page.
No, I
The
mean
page is
the
not
Riverview page.
No, it's not Riverview. This page is not for Riverview. The page is for the centers that I'm handling. I'm handling 7 centers, and I want everybody to be on that particular platform. So if you are not there, contact Esme.
Okay, please, I didn't know that.
She will add you to the platform. And Esme, I want you to send the link to each of the course rep in all the centers so that they can put it on their platform for their group members to join. Esme, is that okay? Or she's not here? If you still insist you want
Sir,
to take the
please,
number, I think this is mine.
I'm from Mauli Center. I don't know whether I'm part of. I don't know whether I'm part of your the centers, the 7 centers you mentioned.
Oh, so you have not checked which particular lecturer you belong to. So it means
I know, but I decided to join. I'm not lost. I decided to join Mr. Boyce.
If you decide to join, then you don't have the right to come and ask whether you belong here or not. That one day I might not be able to tell you. This is the number. Those of you that you want to call. Because every center has been given specific lecturer to handle. Is that not so?
Yes,
sir. Please, I'm aware about that.
I know I am handling the Bolga people. I'm handling Wa. I'm handling Tamari. I'm handling Techimai. I'm handling Riverview. I'm handling a Holy Child. Then 6. It left with 1. I don't know the one is Mauli or what.
Sefi. Master is Sefi.
Good, Sefi. So Riverview, you are not part of my class. You are missing.
Sir, please, the number.
The number is on the screen. Have you seen it?
Hello.
Have you seen the number? It is on the screen.
No, please, I'm not able to see the screen. Yes, please.
Hey. And your phone, there is something wrong. The number is 0246.
Okay.
0246 69 6759.
Okay.
0246 69 6759. Mapon.
Thank you, sir.
You are welcome. Is that all? So I think we can call it a day.
Yes, please.
We can call it a day and you prepare for your next class. If you have read the course slides, get your mobile phone, go online. You can go YouTube and watch some videos to help you understand things better. But if you still don't get the concept, you just WhatsApp me. I'll respond to your messages or your call. All right, Esme, make sure everybody gets onto the platform. Very, very important.
Yes, sir.
I don't want to meet you next week for some people still to complain that they are not on the platform. Right.
Yes, sir.
Okay.
Thank you, sir.
You are welcome. Have a good day. Bye.
Same to you, sir. Bye. Yes. Yes. Hello, colleagues. Good afternoon. Please, anybody, anybody who is not on the platform, kindly see your course rep at your center and then give your number to him or her. I have given the links to them. Some they gave me hi and then I gave the links to them. If not, you can also hi me and then I'll just send you the link. Then you just come to the platform. When you come to the platform, please just write something. Can you please put on the slide? We don't have the slide or something like that. But if you keep mute, we might take it that a colleague has given you the slide, so you don't need it anymore. So please, when you come on and then you want the slide, kindly write something so that those who have the slide will just post it there. We saw people asking and then most of our colleagues were just posting. So if you don't have it, just say something. We'll just repost it and then you will have it.
prefer to see
And then buy a net. Mapo. Answer. I'm saying I'm for about 2 fridges.